Theoretically, as medicine, it's almost mythologically compelling because all cancer cells are different than healthy normal cells. Otherwise, they wouldn't be cancer. So if you can train the immune system to recognize those differences, then every cancer carries within itself, in its genome or in its transcriptome, the seeds of its own destruction.
Today, I'm in beautiful North Carolina, sitting with Alex Rubinstein and Ben Vincent, the two PIs who together run the Personalized Immunotherapy Lab at UNC Chapel Hill. They are amongst the few academics I know who operate at the intersection of cancer, machine learning, and aggressive clinical translation. And I learn something new almost every time I talk to them. Today, we're going to talk about cancer vaccines, neoantigen identification, the cancer care of the ultra wealthy, and a lot more. Thank you both for coming onto the podcast.
Yeah, thank you for having us.
Thank you. It's fantastic.
So the obvious first question is that cancer vaccines are very zeitgeisty subjects right now, like with like Rosie's dog and Sid, the founder of GitLab's his whole cancer journey. And I don't think I quite naively understood what cancer vaccines even are until I talked to both of you. Just for background context for the rest of this episode, could you walk me through what exactly are cancer vaccines and their historical progression over the last 2 or so decades?
Sure. Yeah. So it's an exciting therapy. It completely nerds me out of computer science and into biology. And so the basic idea is that you want to reprogram the immune system to recognize a cancer and distinguish between cancer cells and healthy cells. And that reprogramming happens by just giving examples of, okay, this is something that's bad. Hey, immune system, if you see this bad thing, you should kill it. And if that works, then you don't need, it's a sort of easy living drug. Your immune system now recognizes cancer as distinct from healthy. It kills it on site. That keeps happening, and you don't need ongoing treatment. And it works through a mechanism we already know is effective and safe. We can clear viruses. We can clear, pathogens of various sorts. And so we just treat the cancer as a pathogen, and it's gone. It's a very nice idea. And I think that people recognize the potential of that general trend way before we had the knowledge and technological components to realize it. So it's been kind of a long road to figure out what do you need to do to start making that actually clinically effective. Do you agree with that description?
I do. And I think theoretically, as medicine, it's almost mythologically compelling because all cancer cells are different than healthy normal cells. Otherwise, they wouldn't be cancer, and they can't be cancer. So if you can train the immune system to recognize those differences, then every cancer carries within itself, in its genome or in its transcriptome, the seeds of its own destruction. That this kind of antigen-directed immunotherapy may be a general solution for cancers across cancer types and clinical contexts.
And so like the typical construction of a cancer is that you want to include some fragments of whatever makes a cancer cancer alongside an adjuvant that like alerts the immune system to get these antigens, pick them up, present them to the T cells, and then mount an immune response to the cancer. The obvious question is, what should these antigens actually be? And it seems like you have a really, really good presentation about this, Alex, where you talk about like how the cancer vaccine, the actual cancer vaccine pipeline. And it seems like there's kind of 2 directions you can take. You can find proteins that are typically expressed by cancer cells or expressed outside their usual place. Or you can find what are called neoantigens. So antigens are uniquely presented by presented by cancer cells. I think the cancer field for like pre-2014 was very focused on TAAs or tumor-associated antigens.
Mm-hmm.
I'd like to get like your take for like what this field looks like. And I think like I think if you look at where most cancer research is today, it seems very focused on neoantigens. But I think you make some like pretty interesting arguments that TAAs are actually very useful, and we should be paying attention to them more.
Yeah. And so maybe one thing to start with is, why did you have a progression where we focus on one thing and then we switch focus to the other thing? I think it's not really about the intrinsic properties of those possible targets. It's really a technological shift. The Illumina HiSeq sent everyone looking for cancer mutations. And once we could find them, a couple labs really quickly started figuring out, we could take this whole framework of existing cancer vaccines load them with mutations. That sounds better. It sounds more tumor specific. And so everyone just said, okay, great. Now we're going to do mutational targeting in our vaccines. But if you zoom out, you don't really care exactly how you found the thing. The range of mutations is bigger than what you could easily see with short read sequencing. And so, neoantigens are actually a really big category. We mostly look at just a couple types of those. And then the expressional weirdness of a tumor is also really, really big. Tumors do all kinds of weird stuff. They reactivate endogenous retroviruses. They splice incorrectly. They express, silenced, like reproductive associated genes all the time. They express just intergenic regions of the genome that aren't really protein coding genes, but sometimes create weird repetitive elements. So there's a lot of ways that a cancer is bizarre. And I think all you really care about from the point of view of killing that cancer cell is having something distinguishing that's tumor specific, which is presented, that the cancer cells are going to all be making and presenting that target. And so the distinction, when you get into any kind of people trying to operationalize cancer vaccines, they treat these as like really different beasts. They're like, oh, are we going to do a TA vaccine or are we going to do a neoantigen vaccine? And I think it actually doesn't really matter. You just want distinctive tumor surface targets, which from the point of view of vaccines, we haven't talked about the immunology. We should.
Yes.
So when I start saying MHC presentation, and some of the audience is going to feel lost, but what you want is things on the MHC of a tumor cell that are not on the MHC of any healthy tissue that you care about. So that can be an expressional target. It could be a protein like PRAME that's really reproductive in function. It can be a virus. If you have a viral origin for your cancer, HPV is a great target. A lot of the cancer vaccine success stories are with HPV. And it could also be mutational in a really broad sense. It could be a splicing error. It could be all sorts of stuff. So I think as we move out of the sort of exploratory tech build phase of cancer vaccines towards clinically effective ones, all this stuff is just going to collapse into find all the targets that are actionable in the sample and then prioritize those as a unified set and pick the best ones and go after those.
It's probably important to say that it's highly likely these will be largely individual person to person. So if you have, all pancreatic cancer patients whose cancers, have a certain, say, RAS mutation, which is very common. Only a minority of those people will have the right HLA and then to even potentially present a certain mutant RAS peptide. Or, if they have the right HLA to present it, actually have the protein processed and have it presented at enough copies of the peptide MHC on the cell surface to be recognized by T cells.
So maybe we do talk about the immunology and like define.
I think it's worth doing like the little bit of background.
Yeah. So you have let's define the words MHC, HLA, and perhaps like neoantigen.
Okay. So Ben, you should do it.
All right.
I can feel myself wanting to go deep in the weeds. I feel like Ben is good at staying on task.
So, cells of the body all express what's called, class 1 MHC. MHC stands for major histocompatibility complex. HLA stands for human leukocyte antigen, and HLA is the MHC of humans. So you'll hear us use those terms interchangeably, but really HLA is the MHC in humans. And those are molecules that bind to short peptides and present those peptides on the cell surface to then be recognized by T cells. And so there's actually two classes of classical MHCs. Class 1 presents short peptide epitopes to CD8 positive T cells, which are the cytotoxic T cells or the soldier cells of the immune system. Class 2 MHC presents a little bit longer peptides to CD4 positive T cells, classically known as helper T cells. But there's some reports now in cancer that those can cause direct cytotoxicity, just like CD8s. Immunology is very complicated. That's part of what makes it so much fun. But in order for a peptide to be presented to a T cell, it has to be derived from a protein that is within the cell that's then processed, chopped up into small pieces. Then those small pieces get loaded onto the MHC molecules and the peptide MHCs pairs, complexes get shuttled to the cell surface. And there's sort of complicated ways that happens for both class 1 and class 2, but that's the basic principle. So T cells can only see cancer-specific aberrant peptides if those peptides are actually bound to and presented by MHC molecules on the cell surface, or in humans, those are HLA molecules on the cell surface.
And there's also this, maybe this is wrapped up in what you just said, but there's also this, like there's heterogeneity in how the MHC is structured from like ethnicity to ethnicity. And I think there's even like subdivisions within that.
Yeah, this is super important. The MHC or the HLA loci in humans are the most polymorphic of all of the germline encoded regions. And there's huge differences in, HLA allelic distributions by ethnicity. And how many are there unique now, Alex? 20,000 plus?
I've been curating pan species, but I'm up to like 60,000, but a lot of that is human. Like there's a lot of human alleles, like maybe like 20,000 human alleles known. Every time you find like a new, like remote mountain village, you'll probably find a new HLA allele.
And this actually really matters a lot, because what the T cells are recognizing is not the peptide epitope by itself. It's the pair of the peptide epitope and the MHC molecule that presents it. And that's one of the main reasons why the same mutations in different patients will be or won't be presented, because if those patients have different MHC allele haplotypes.
So in an absolute ideal setting, you have this cancer cell that has some sort of MHC on the surface. It's expressing the aberrant, like chopped up peptides that it's creating. A T cell wanders by. The T cell has gone through like a self-selection process. So like it's looking for things that are like foreign to the body. It looks into the MHC, it sees something is off, and I think it either instructs the cell to kill itself, or it just kills the cell.
That's right. That's roughly correct. There's a few things I want to say about that. One is that the more familiar place where we see this act is in viral infection. So like, why do we have this entire like papers, please surveillance state where T cells are going around getting samplings of what cells are making and then killing them if it's wrong? It's really, I think, evolutionary geared towards viruses. A bunch of weird proteins show up in a cell, and then some T cells show up and they kill it. And so this is kind of the like clearance phase of a virus. There's, I think, only so far you get with just neutralizing antibodies. I think that beyond that, you need other mechanisms. And so some of those mechanisms are the second adaptive bit of the immune system that can sample the interior of a cell and then kill the offenders. So I think that should be at least a starting mental model for like, how do we even kill the cancer cells? Well, you get them to get cleared the same way that virally infected cells get cleared. That's one thing. Another thing is just numbers wise, it's useful to think about how many MHCs are in a typical cell. And like, I often just kind of come back to this, and it varies a lot, but like around 100,000.
Now that you brought up this, I kind of assumed it was one, but it makes sense. It was many.
Well, so you kind of, it serves information theoretic purposes. You're trying to, there's an infinite diversity of stuff going inside of a cell. And then you want to get a really succinct kind of histogram of all that activity. And so if every single subsequence of every protein were on the surface of the cell, that's just like way too much. Like you can't lock onto a signal. If it was just like one, single MHC, first of all, how would a T cell even find it? You have to like scan the entire surface looking for the one MHC. So you need a bunch of them. So when a T cell comes by, there's a high probability that there's an MHC for it to look at. And then you need like restricted, only very specific subsequences of each protein get onto those 100,000 molecules. Because if every subsequence of a protein could get on there, even if it's, there's 100,000 and you can look at one and it's some actin peptide, but it's a different one every time, you can't lock onto like, okay, wait, is this the weird one? Or like the information is just diffusing. So you need it to be really repetitive. Every protein is represented by a few exemplars. And so you achieve that through a few things. One is that MHCs are a kind of delightfully easy machine learning problem, like which peptides they present is really easy to predict. And so the biology of it is very restrictive in sequence space. They have strong preferences like position 2 needs to always be available, things like that. And then the antigen processing also kicks out a lot of things that could encounter MHC. So it's this like 100,000 molecules on the cell surface, of which maybe, there's 10, 20,000 distinct peptide sequences. And that's tractable for T cells as a distributed machine learning algorithm to process. Does that make sense?
It does make sense. I think an instinctive question I had when you first explained this to me is, if the MHC, these 100,000 MHC molecules, or how the T cells are like looking into the interior of a particular tumor or a healthy cell, why doesn't the cancer cell just drop its MHC? And
So they do sometimes.
Yeah.
So there are a variety of mechanisms that keep cells from doing this, and I think for reasoning about that, you should think more about viruses, because viruses often try to like hijack and subvert and repress MHC.
Yes.
But there's a whole other compartment of the immune system called NK cells, natural killer cells, and their job is roughly to make sure that cells are presenting something on MHC. So it's not like a perfect pressure, because cancers do eventually find their way to dropping MHCs. At least certain cancers in some settings do. But there's some evolutionary barrier there. Like if you just naively do it, there's a whole other cell type that comes and kills you.
Gotcha.
There's also the MHC locus is pretty dense, and it has some things that are not related to presentation of T cells in there. It has other like RNA polymerase genes and things that if you drop them, you also suffer a hit in fitness. So if you look at cancer sequencing data, especially if someone gets checkpoint blockade and you know that the T cells were doing something, but then there's an escape clone that is the recurrence, often there's a loss of, a part of the MHC locus. Maybe a particularly important allele that was presenting something that the T cells really relied on to know that cancer was a cancer. That kind of stuff, chopping off like a bit of a chromosome to lose an allele or two is easier than losing both copies of the full MHC locus. It still does happen, but it seems to also open up other vulnerabilities. And one of the craziest things, and I like this story and understand it, is that microsatellite unstable colorectal cancer often ends up with some or complete MHC loss, and then is really sensitive to checkpoint blockade. And like totally breaking the entire mechanistic diagram. And so there's some papers that came out that are like, oh, gamma delta T cells, which you normally don't think about, they pick up the slack and they do it. But the details of that are not worked out. But I think the thing that is that, just losing MHC does not make a cancer invulnerable.
Gotcha.
And even cancers that, there's a few transmissible cancers, and in their transmissible state, they lose MHC, but through repression. And so there's like a nasty like dog STD cancer. When it's in a new host, it has to start making MHC again for some not worked out reason related to fitness, but like it's just not trivial to completely get rid of it.
Is it fair to say that if I was a cancer cell and I wanted to evade the immune system, I feel like the obvious thing I would do would be to continue like presenting normal looking peptides onto the MHC? Is it just like the act of becoming cancerous is so orthogonal to that just doesn't really happen?
That's actually one of the things that makes cancer cells harder to see than virally infected cells.
Okay.
Than virally infected cells, where they're pumping out huge amounts of the viral proteins, there's a much more higher density of viral peptide specific MHCs on the cell surface. In cancer, the vast majority of the MHCs are presenting self-peptides.
Yeah.
So it's the aberrant peptides are hidden amongst the sea of self-peptides, and that makes it more difficult. But the other thing, we know there's selection that works along that axis, because you can follow tumor subclonality developing over time, and you can see certain subclones drop after immunotherapy or under selection pressure. And then the cancer that grows out, maybe there was a mutation in a passenger gene that it didn't really need for fitness, but there was T cell selection pressure through that, and the cancer will just drop either expression or lose the genetic material. So it's no longer vulnerable to those T cell populations, which incidentally is one of the reasons that Alex and I think we really need to be doing immunopeptidomics validation of peptide presentation, because if we're giving vaccines to peptide antigens that are predicted but not actually presented on the tumor cell surface, we can be eliciting immune responses that are completely useless to controlling the cancer. They can be real and measurable by immunological assays, but do nothing clinically.
And so just to define immunopeptidomics, it feels like there are two options on the table if you want to learn what peptides is my cancer cell actually expressing on the MHC. One, you can do like whole exome sequencing or whole genome sequencing of the cancer, like identify where are the mutations, and then like slide a window over that region to create all the possible peptides. This, I think, is a partial answer to the question.
That's like candidate sourcing. Like there's other ways you could do the candidate sourcing. It doesn't tell you what's really there though.
Yeah. And
I guess if your informatics got good enough, you could start squishing like in my mind, it's like the generation of the candidates is really different from validation because the candidates are so low value. The predictions are so bad. But if you got really good at the predictions, then maybe you could start to conflate those two more.
So I actually like from looking at the clinical trials that have been ran with cancer vaccines, they do seem to be like sourced directly from the genome, and they don't actually tend to do this like to actually define immunopeptidomics. You actually like lift off the MHC, run it through mass spec, and identify all of the peptides that are on that region.
Yeah. Okay. So let's talk about how you'd validate, and then I'll tell you why no one does it. So if you wanted to know what's actually on a tumor cell, you have a few options. You don't have to do mass spec. Maybe I'll start with the other alternative that like is even rarer, but a few companies have wandered into. You could look at tumor infiltrating lymphocytes. So you look at T cells in the tumor, and if you you'd sequence their T cell receptors, you could do some work to figure out what do they recognize, and you could also look at like what was their sort of expressional state when they're in the tumor. And there's certain signatures that are related to like, hey, I found my target, I'm going to kill it now. And so you like focus on those clones, look at their T cell receptors, and then take your whole candidate set. You do your typical thing is you do exome sequencing and RNA sequencing, and then you run that through like UTECT and Strelka, and then you run the predicted mutational sequences through like NetMHC-Pan. So you take all that stuff, which is a really long list, almost none of which is on the tumor. And then you do some work to figure out the T cells in the tumor that look like they're dividing or trying to kill stuff or just generally activated. Like, do they recognize any of those targets? And if you find T cell clone those in the tumor that recognizes a predicted target, that's really strong evidence that probably the tumor was making it. It could be that you screwed up the matching of the T cell receptor to the antigen. Maybe it's cross-reactive. You need to do a little bit more work to get confident, but like that's already way more validation than these sort of just predictive searches give you. That's one way you could do it. A totally different way is if you have abundant frozen tumor tissue. This is like step one of why no one does it, because all of the logistical machinery of a hospital goes against doing this. They don't want to have freezers full of stuff. They want to have room temperature, formalin fixed, just a little bit of tissue. They don't want to have like, frozen chunks of tumor in freezers from every single patient. But if you have a lot of frozen tissue, then you can kind of hook into mass spectrometry to detect some of these peptides. And there's different ways to do it with different sensitivities. But the basic idea is that you're going to pull the MHCs off the cells, and then you change the pH to make the peptides pop off. And then you drip, you put them through a column so that you get this gradient separation. And then you run it through, often you'll do something like Orbitrap you do like 2 stages of, you figure out like, okay, what's the mass to charge of the peptide, and then you fragment it, and then you look at the mass to charge of all the fragments. And you can informatically figure out which peptides were coming off the tumor. So this is like a lot of work and also requires a sample that's pretty much never available. And if you do that the way I described, it also has a lot of sensitivity problems. So you like actually can't see a lot of the peptides for a variety of technical reasons that we can talk about if you want. So if you really want to know what was there, you have to do a whole extra thing, which is you So like naively, you could, one of the problems is that mass spectrometry has severe biases in sequence space. And so if you wanted to know, like, okay, but where should I point the acquisition of this instrument and what should I expect from the peptides? Not from this like, computational figuring out from lots of spectra, but just I really want to know. You could make some of the peptides, synthesize them, and then you could kind of try to figure it out. But it's still like that's not quite right. It's like it gives you a little more information. So there's a trick. You could make the peptides, but they're made in a way that they're heavy isotope labeled, and you could drip them in with the peptides from the tumor, so you know exactly when to acquire. And then you also run them separately, and you know everything about like, okay, this one doesn't really fly so well, but we're still going to see like it's going to at this moment coming off the column, you're going to see B's peaks. And so you combine the like information about the reference peptide with the fact that it's running with your sample.
Mhm.
The reference peptides cost like $1,000 each to make and take like a month, right? So it's this whole extra, like now you want to check 100 candidate peptides, it's $100,000. And it's an extra month before you do your mass spec, which is a complicated, fancy assay, before you start making your vaccine itself, which takes another like 2 months. So like, I wonder, is that enough to explain why no one does it? Or like,
Okay, like so, I guess like maybe if you were trying to be like maximally accurate, you would need to go through this like whole isotope labeling thing that
Mhm.
is super expensive and super like consumes a lot of time. Alternatively, it seems like what you get out of the mass spec is perhaps not all of the peptides, but at least the peptides you do get, you're confident about. And maybe is that enough?
right. So you can run it in different modes, and there's ways that have more sensitivity. And so you could theoretically try to do mass spec in a way that's like, we're accepting we might only catch a quarter of them, but those are still actionable. I think that people did try that a bit. So Michal Bassani-Sternberg, who's a like great mass spec immunopeptidomics researcher, published a lot of work on this, trying to read off mutational targets from melanoma samples. Because melanoma, there's like, big cutaneous chunks of tumor. You can get it fresh. You can like re-biopsy if you need. So like it's a more accessible tumor type. And so she did a study in like 2016 where she tried to find these neoantigen targets on 25 samples, and she found them on 3 of the patients. Coincidentally, what she did find a ton of is me and Ben's favorite target set, which are cancer testis antigens.
Sure. Yeah.
And we like them because they're, you can look in public data and know that they're real. And so it's like, we can kind of come back to like, what evidence would you want to fix something? But, so she did that in 2016. I don't know if she's done a repeat of that. I think if you, with a little bit of update on the methods and newer instruments, because there's now like fancier, more sensitive instruments, you might get that up to like you find a peptide in like 10 of the patients or something. But it's still limiting. I think without doing the really annoying targeted version where you can quantify and get down to really low abundance, you end up sometimes just having nothing.
Yeah. And in that case, you might as well just opt for like the whole exome sequencing approach.
Well, you still do the whole exome sequencing.
I mean, you do
do
like
that.
a confirmatory.
Yeah, like you have to do that in order to even know what you're looking for.
That's fair.
You can never get away from it. Like if you want mutational targets, you have to do some kind of sequencing. You could do, it doesn't have to be whole exome sequencing. If you did, just a single cell long read RNA sequencing, we have like a method that might work for that. But like, you still need something that's nucleic acid from the tumor generating a candidate list for you.
I think one of my favorite papers in this space, X et al. From Claude Pro's lab and leukemia from 2021, they went very deep and included using a genomic reference against expressional antigens that aren't mutations, but are, part of the dark proteome, so to speak. And their findings were dominated by tumor-associated antigens. Again, I think it was a really, really well done work. And maybe the answer is, the answer to your question of, is it enough, right, is conditioned on how many do you need to make a good vaccine. You need at least one that's actually presented by the tumor cells, right? But you probably need more than one because of the potential of HLA loss. So if there's two copies of, the HLA locus on chromosome 6, and it's relatively easy for the tumor to genetically lose one, then ideally you would make a vaccine that has epitopes that span HLAs on both chromosomes, right? So that then creating selection pressure that would cause the tumor to have to drop both and incur the fitness costs therein rather than just dropping one.
And ideally spread across different alleles because when the loss happens, there'll probably just be like a piece of this one and a piece of that one.
I was going to ask, like, I feel like a lot of, like maybe this is incorrect, but it feels like a lot of cancer vaccine people just kind of implicitly assume like this patient has this particular MHC allele, and they just design, they pick the neoantigens using that as a reference point. But are the tumors able to modify their own MHC?
Like mutations in the MHC coding regions?
Yeah.
I think people have found that, but it's really uncommon. I mean, I think that like this sort of, like when you think about tumor evolution, you got to think about like what's the diversifying mechanism? Like by which means can tumors have a big population that then evolution acts on? And there are some that have infinite point mutations, but they're more commonly can diversify by just like losing chunks of chromosomes.
Yeah.
And so I think in a cancer that's driven by some mutational process to just introduce tons and tons of point mutations, that one will stumble on like, oh yeah, you should disrupt the, residue 72 of the binding groove. So, but like, that's a hard trick to pull off for a cancer that's not like point modifying everything in the genome.
So we've mentioned the word TAA. We've also mentioned the word CTA, cancer testis antigen. I think I would love to get your explanation of that because I think it is a very like fun and clever way to approach cancer vaccines.
Yeah. Okay. So TAA I think is older terminology, and so this is people looking at cancers like 30, 40 years ago and finding that there are some proteins that just come up a lot. And so it doesn't actually say anything about the specificity of it. I think often they're kind of an overexpressed, like, oh yeah, cancers will just make a lot of this protein. And there are different therapeutic approaches towards the proteins that come up a lot in cancer samples, some of which were vaccines. And then there's a lot of other therapeutics that people pursued also. If you wanted a subset of TAAs that had really nice tumor specificity, so you feel no worry in like control after deleting every cell that makes that protein, then you get to something that's roughly cancer testis antigens plus a few other things. But like cancer testis antigens are lineage restricted to some stage in reproduction, and most of them are related to spermatogenesis. So they're like a strange moment in spermatogonial stem cell development where they're not yet pumping out tons of protosperm, but they're about to, and they need this one protein. And so like those kinds of proteins. There's also placental proteins. Placenta is super weird. It does all kinds of stuff that no other tissue in our body does. There's things like, bits of the like motor protein of a sperm that other, cells in our body don't need that. And so I think it's not strictly accurate to say that these are not unhealthy tissue, because if you're a guy and it's like the motor protein of a sperm, a really effective cancer therapy would also probably make you infertile. So in some ways they're like, disposable tissues or tissues you can warn against. You don't want to give a cytotoxic therapy against placental proteins to someone who's pregnant, but like you can kind of navigate around the reactivity to make it not toxic.
I feel like importantly, like the fun part about CTAs is that there is no T cell repertoire, or like, T cells have not been selected against them.
So some of them have. This is an open question for us. So we went looking, and most of them are not expressed in the thymus, which is to my surprise, because they're in the genome. And the thymus, so we don't talk about the thymus, but it's kind of an education. It's like a finishing school for T cells. And they go there to figure out what not to kill. And so most cancer testis antigens don't show up in the thymus. So T cells are not educated to not kill them. There are a few that do, and some of them are like really high value. There's this one that I would love to make a therapy against FATE1, because every Ewing sarcoma makes FATE1. It's like required to by its oncogenic fusion. But that is the one that we saw the highest like thymic expression for. So, the therapy as a vaccine might work, but it's less likely than some other ones that are available that the thymus is just really silent in those.
This is a really important point, though, because the T cells are negatively selected against selfness, not at the level of the complete genome, but at the level of which proteins are expressed in medullary thymic epithelial cells under control of this really cool protein called AIR that causes the MTECs or medullary thymic epithelial cells to be able to broadly express genes from across the genome, but it's not completely uniform.
Without dying, which is weird.
Without dying,
which is the proteins, when they all put them together in one cell, should just it's not any one cell type anymore. It's just a jumble of you're a little bit liver, a little bit brain, a little bit sperm, and then somehow they don't die.
And thymus cancers are very rare. I mean, they exist, but this is crazy biology that has not yet been unlocked.
Yeah, I guess I never naively thought about how the selection training happens, but it makes natural sense that there's a cell in charge of it. It is strange, though, that they're able to express every single protein. Just
super surprising.
And so that in a large number of, as Alex said, a large number of the cancer testis antigens are not expressed in the MTECs. By the way, the MTEC carefully curated RNA sequencing data set we use also came from the Pro Lab. So super thankful for that, to see this. But the famous cancer testis antigens, of which T cells have been discovered, by multiple groups over many years, they're all nearly negligibly expressed in the medullary thymic epithelial cells.
Has that stuff been mapped out? Every possible CTA that exists? Okay.
No, I spent a month this year doing a new curation of CTAs against different data sources, and I got a bunch that I think that the criteria have just changed over time. The available technology changed, and so there are some studies that'll find 2,000 of them. But then if you really do care about, I just don't want any brain expression of this. I don't want to make a T cell that goes in the brain and finds its target there. And then you lose a lot of that 2,000. The brain makes a lot of weird proteins. If you're really cautious about cardiac expression, because there have been CTA targeted T cell therapies that cross-react to the heart and they kill people, then you lose some of that candidate set. So I think that maybe it's not right to say that, you could just go and find a bunch of new ones. I think that the work of finding really actionable ones is still a little bit open.
Why is it not as simple as this particular cell in the thymus contains the full universe of possible CTAs, and then I'll just cross-reference that with the transcript of every other cell type in the body?
Well, so there are good data sources now, the Human Protein Atlas lets you, get many different tissue types. One problem is that there's a lot of further subdivision of, normal encompasses a lot of different cell behavior. So it's not enough to just, it's you got a brain sample, great. Well, you need to treat the cortex different from, the cerebellum. And so then you start these subdivisions, and then someone else goes and they do the single cell sequencing and they find that actually there's 5 different subtypes in this one area. And then you need to do the normalized TPMs within each subtype, because you do get, rare cell types that if you kill them, you give someone diabetes. If you had some broad expression profiling of the pancreas, you would miss the very small number of cells that are pretty vulnerable to getting killed. So, I think that work of defining normal is not exactly finished, even just the level of how much of each gene is made. And then there's a whole nother repeat of that entire unfinished project of doing long read sequencing, because you don't know all the isoforms that they make.
Oh, yeah.
And so if you wanted to capture the real isoform diversity, then you need to go back and redo GTAX and the Human Protein Atlas and all that work with long read sequencing, which I think some people are starting to do, but it's just an unfinished spot in our reference databases.
Now you think
No, just another level beyond that. The subcellular location of proteins matters to how efficiently they're processed and their peptides presented. So take, for example, there's a leukemia antigen that's a self-antigen called cathepsin G. Cathepsin G is essentially in granules that are not normally exposed to the cytosol. So it's not normally presented on HLAs from normal, healthy bone marrow hematopoietic stem cells at a very high level anyway. But some leukemias actually express this and get it into the cytosol aberrantly, and then it's expressed at ridiculously high levels, and it's a target for therapeutics. So it's in the genome, it's in the self-transcriptome, right? It's expressed in medullary thymic epithelial cells. So by all rights, it should be a dangerous self-target. In fact, it's a very fruitful target in at least myeloid leukemias because of this subcellular distribution of the protein and relative likelihood it'll get processed and presented.
Sorry, maybe a naive question. Shouldn't you expect the adaptive immune system to clean it up then?
Well, so
The adaptive immune system like flops all the time. Like it's like, yes, it kills a lot of cancers. Probably I have in my lifetime had some infinite number of cancers that would be deviated cells that got cleaned up. But, there's limits to how much it could figure out.
No, that's actually a really good question though. But because it's expressed in the medullary thymic epithelial cells, the T cell precursor frequency is very low. So you want to target it with something like a TCR-T, a T cell that you exogenously engineer to have a T cell receptor that will bind it that you've discovered some other way, or an antibody or something like that.
But also if you look at we should talk about what are the success stories for T cell mediated killing, and because and if you just look at when have we gotten T cells to really eradicate established cancers, it's a pretty short list, and a lot of them are engineered cell therapies where you skip the vaccine part and then you stick a receptor that recognizes your target directly into T cells. And so that's mostly for cancer testis antigens. So for PRAME, NY-ESO-1, MAGE-A4, these sort of reproductive proteins. Also for viral proteins, so HPV, a few mutational targets, but not a lot. And usually the ones where you had good mass spec evidence that they're presented. And so if you look at those success stories, like if someone has a PRAME positive melanoma, which like a lot of them are PRAME positive, there's this great TCR-T from Immatics. So it's an engineered T cell receptor therapy that seems to work pretty well.
And this is approved.
And it's not yet approved. I think they're having a phase 3 trial right now. But I've heard from doctors who've used it on patients that you get impressive responses. Not always durable ones because it's one target, but impressive regressions of tumors. Those tumors had PRAME all over their surface to begin with. And PRAME, and when we look at the MTEC RNA-seq, the expression 0.05 TPM, right? So there's not a lot, maybe somehow there's a lot of tolerance being induced there, but probably there's not a lot of tolerance. And so the capacity to kill the cancer mediated by this one PRAME-derived peptide was in the patient's immune system the whole time. But immune systems just try to do a lot.
Yeah.
Just because there's one good target in a cancer does not mean that when the cancer cells are, imploding and draining to some lymph node, that from the debris of the cancer cell, the immune system will just figure out, aha, this is the good target, we're going to go after it.
I also imagine that you basically mentioned this, but the adaptive immune system is also dealing with the fact that the cancer is actively suppressing it.
Yes.
And I do wonder, how much of the pre-Keytruda cancer vaccine literature was actually hampered by Keytruda not existing, and if we went back to it, there would actually be a lot of really rich stuff there if you just combined it with Keytruda.
Hopefully that's true, but the PD-L1, PD-1 axis is not the only axis of immunosuppression. So, one of the things I think about a lot is, I think, we're talking now about how to personalize vaccine or, maybe TCR-T therapies. I think the reversal of immunosuppression and immunoevasion mechanisms activated by the cancer will also have to be equally personalized for every patient. So for some people, Keytruda or similar is going to be the right thing to give with the vaccine, right? For others, it's might be a thing that eliminates regulatory T cells or myeloid-derived suppressor cells or tumor-associated macrophages or cancer-associated fibroblasts, or the cancer's ability to actually upregulate molecules that kill the T cells that come to killing the cancer, right? There's so many different ways that the cancer can fight back against immune recognition and killing that I think we're fooling ourselves if we think there's a one-size-fits-all solution.
That makes sense.
But I think that my hesitation around like, did all the old cancer vaccine literature not work just because we didn't have Keytruda? And there's a few things there. One is that I have like a personal annoyance to the degree to which cancer vaccines hide behind checkpoint blockade. I just add it to every single trial, and then you get like essentially the expected historical response rate to checkpoint blockade. Then everyone looks at it and says, "Aha, the cancer vaccine worked." So like, yeah, maybe add Keytruda, it would work. But like,
They measure vaccine-induced immune responses.
they do in the trials.
Yeah.
You can talk about that. But I do think that like if that were the issue, it wouldn't be uniformly the issue. And so in those trials, you would see a few exemplars of where the cancer didn't figure out, oh, you should make some PD-L1. And then those people would have had strong monotherapy responses to the vaccine.
Mhm.
And when you look for monotherapy responses to the vaccine, even in like a small fraction of a trial, it is such a short list. It's like HPV vaccines in really early stage cancers. Like there, you can really point to like, oh yeah, there was a really early stage or sort of precancerous condition that's HPV positive. The vaccine could clear it sometimes. There's a BioNTech melanoma trial in which they had one complete response. But to me, like as someone who works on cancer vaccines, I think BioNTech did not see it as a success. It's a low response rate. They added checkpoint blockade. It only boosted it a little bit. So they, I think that they kind of shelved that fixed antigen vaccine. But I saw their phase 1 trial and thought like, holy shit, they did it. Like they, it's only one patient, but like no one else has done that. Yeah. They didn't need some combination that sort of obfuscates, exactly which agent's doing the work. They gave the patient a vaccine. They got like 15% of their circulating CD8 T cells recognize antigens from the vaccine. And then their melanoma disappeared. That is a setting in which you can then go look at the other patients and see, okay, well, who else got like 5+% circulating T cells responding to the vaccine who didn't get a complete response? Let's look at their cancer and see, was checkpoint blockade the missing part? And I think that's why in a subsequent trial, they added a checkpoint blockade agent. And it didn't work as well as they hoped. And then they got, they threw a small fit over it, I think. But like, maybe the answer is not easy, but that's where you need to go look for what is the extra thing that's missing. You need to have a strong success signal, even in just one patient, to then figure out, okay, like it's the potential's here, so why didn't it work for the other people?
We can come back to the actual like neoantigen or CTA part in a bit, but yeah, one thing you did mention of like maybe we just cut out the middleman here and we just engineer the T cells against the particular antigen that we want to mount an attack against. Why doesn't everyone? Is it just expensive? Is it logistically complicated?
It's so expensive and it's so complicated. So we had that thought, and so we've gotten into doing TCRT engineering preclinically in the lab. We've been working with a sort of rebooted, personalized TCRT company. We've tried to like make broader contacts in that space. There's a lot of reasons why that's not like the current go-to solution, but I think the simplest one is just how logistically complicated and expensive cell therapy manufacturing is.
Mhm.
So it is really quite a slog. And you can kind of see it with CAR T because CAR T felt like a miracle. And then the deployment of CAR T is really muted. Like not that many people get access to it, even though it's like a single construct. Like you're just trying to get this one construct into people's T cells and give it back to them. And it's still like almost a civilizational challenge to scale it up. So imagine you're trying to do that with a set of different receptors per person and you have to find them first. It's really hard.
Yeah. It's going to happen though. I mean, it's part of what we need to cure otherwise incurable cancers, I think.
I mean, Ben and I are
We're gonna
working
get
on
there.
that. Like we are actively working on, faster ways to do, like in vivo TCR therapies. But there's technical challenges there. And I think in the meantime, there's kind of a middle period where, we have hints of success from the vaccines and they're logistically much easier.
Mhm.
So probably it's a reasonable place to allocate like 80% of the effort there while you try to bring up the technology for the more like higher impact, more effective therapies.
I imagine the concern, and maybe this is not a consistent concern with both CAR T and also especially TCR is that sometimes you get these like weird cross reactivity things that you couldn't have predicted in advance. It was like one of the things I stumbled across was like this like MAGE A3 age site reactivity. Yeah, the PTA vaccines were not working, so they're like let's just make the TCR and cause
Really high affinity.
cardiotoxicity. In some sense, like it feels like if you cut out the middleman, you also cut out like some safety features, and that you might design something that is just very toxic to the patient. Is there just like that's just like you have to live with that?
Yes.
Or
Well, one nuance there is where do you source the T cell receptors that you're going to deploy as TCRTs?
Mhm.
Because if you source them from the individual patient that you're then going to dose, then all of the TCRs that you discover that are antigen specific have been through thymic selection in that individual. So in that individual, they've been at least safe enough not to cause, fulminant autoimmune disease before you gave them back to the person.
but I thought you're engineering those.
Well, that's your other option. Yeah, your other option is to pick them allogeneically, right? And then you could ex vivo engineer them for better affinity. And it's the affinity engineering that I think introduces more potential cross-reactivity danger without an effective screen than sourcing them autologous.
And there's a preprint that just came out. That's great. They're looking at that TCR, the A3A TCR that I think killed 2 patients. They dosed them on the same first day of the trial, which is crazy. And they looked at the affinity enhancing changes that were made to the TCR and then rolled them back one at a time to see, can you get rid of the tightened cross reactivity? So it'd be cancer specific, but not heart specific. And they can. They got rid of 2 mutations. It was surprisingly in like not the loop you expected, but the second loop is in CDR2. And they're able to make a TCR that was higher affinity than the parental, like TCR that had been taken from a donor, but was not reactive with the heart. So like that work of affinity enhancement without introducing cross reactivities is really, really delicate. And Ben and I don't want to engage in it at all because it seems like very stressful. But there's an opposite extreme that Ben was talking about. You can source the TCRs from the patient. There's some challenges there. You're adding a bunch of extra time. The turnaround time of like the needle to needle time here is getting kind of long because you have to do TCR discovery in the patient before you then put the T cell receptors back into their T cells. But that has a much nicer safety profile, like the safety logic is better. And then Ben and I have also talked about a kind of intermediate possibility that if you could do really large scale T cell receptor discovery against shared targets, like cancer testis antigens, you could have a big database of T cell receptors that, you have specificity to a CTA target and the HLA type of the person that they came from. And you can kind of like you do the way that you match people like as tissue donors, like, oh, you have a 4 out of 6 locus match and this TCR will hopefully be safe for you. So, and I think that would be kind of an intermediate amount of risk. You could still potentially personalize, but because it's not heavily affinity enhanced, then you're less likely to just introduce a crazy cross reactivity.
Has any of this reached? Like I know there's some CAR Ts that are approved. Has any of the TCR stuff been approved? Like personalized?
Yeah. So Adaptimmune, they got one approval and they got like a breakthrough designation after their approval. But this kind of comes back to how hard these things are to manufacture and how expensive. So Adaptimmune made 2 TCRTs that work really well in rare sarcomas for which you really need therapies. And they hit CTA targets and they're, get high response rate. They're great. They then went totally bankrupt and like had to sell all their assets to some other company because the manufacturing build out just ate all of their money and their expected reimbursement for these rare sarcomas was kind of low. So they just ran out of cash. But they got it there. There's an approved therapy, and probably Maddox will get theirs approved. And the ones that are, the ones that are going after that category of targets look like, as a class, they look reasonable. Like probably there will be more approvals than that as time goes on, but it's really slow to build them out.
And so, like I imagine, the obvious ideal world solution to this is just manufacture them inside the person. How good is in vivo TCR today?
Uh, I mean, it doesn't exist. I think we're doing it in our lab to some degree. Like
In vivo CAR T.
in vivo CAR T exists. So in vivo CAR T exists. So Capstan has one in vivo CAR idea. Okay, so we should talk about what in vivo CAR T means.
Yeah.
So in vivo CAR T is some version of we're going to skip cell and gene therapy manufacturing, and we're going to instead inject the patient with something that will either in some way get the receptor into their T cells, either permanently or temporarily, but that CAR is going to be in patient T cells.
Right.
And so then the manufacturing profile for that is really comparatively simple. Like at the worst, it's like a viral vector. The easiest versions are like it's an RNA. And that is that's a bubble right now. Like there's a lot of players that have entered that space and more that are rushing in. And some of those have gotten to the clinical stage, and there's there's big acquisitions happening, and they seem to work. They have maybe different properties depending how you do it. So Capstan is doing an RNA-based one, and we're kind of inspired by them to then do RNA-based TCRTs. And then there's a Chinese company, Magic RNA, that has something kind of loosely similar to Capstan, but they got clinical data way faster. But they used theirs actually for autoimmunity. So they nuked B cells temporarily to get rid of lupus, and it seems to work.
Interesting.
And so there's clinical proof there. And then there's a bunch of, kind of riskier, but more effective for cancer versions of this that are actually integrating in some way. And they create stable CARs that, the RNA's got a burst of expression and then it goes away. Whereas if you have some sort of viral vector, you can actually make actual CAR Ts and then they stick around.
And a few important considerations there are, one of the limitations of most of the in vivo methods is you can't use lymphodepletion first, because if you deplete all the T cells that you could then be in vivo engineering, it would be counterproductive.
Mhm.
But the dogma goes back to the early days of cellular therapy that one must have lymphodepletion for cellular therapy to work. I don't necessarily believe that's true. I think one must have means of the cells in the cell therapy expanding and persisting and doing their job. Lymphodepletion is one way to affect that, that we knew 30 years ago. There are other ways that we can, achieve that same purpose now, but that's a hypothesis. And there are some companies that are working on ex vivo scaffold technology, where you sort of have a scaffold that's impregnated with T cells, and then you give lymphodepletion, and then you can implant the scaffold back in. So then the T cells will release, so that you can get sort of the best of both worlds, the manufacturing advantages of an in vivo solution, but also still be able to give lymphodepletion. I think the degree to which we need lymphodepletion is really important in which of those strategies.
But if it turns out that if you can do it without lymphodepletion, then that is the ultimate patient win.
Yeah.
Because lymphodepletion is hella toxic, very dangerous,
And that's literally sifting the T cells out of you.
No, lymphodepletion is chemotherapy.
Oh,
So
okay.
preconditioning chemotherapy. So person has cells manufactured. Before those cells are given to that person, they get highly lymphodepleting chemotherapy regimen, so that, the majority of their lymphocytes are gone.
Sure.
And then the T cells are infused so that they'll be, what's commonly, taught is there will be immunological space. And what immunological space is some combination of physical space in the secondary lymphoid tissues, availability of antigen presenting cells, availability of cytokines and chemokines that the endothelium may make, because there's not a lot of T cells around.
Your body has a set point for how many T cells it wants. So if you get rid of all of them, it starts cranking out all these signals for T cells to grow. And that's when you infuse CAR T.
So the infusion, expands more in vivo, because there's this physiological space for it to move into. But the lymphodepleting chemotherapy is actually a fairly intensive chemotherapy regimen. It means you often can't treat people who are older with serious comorbidities, or even when you do, there's risks of infection and bleeding, other complications, organ dysfunction.
People just die of it. I mean, our T trials, there's they look at the mortalities, it would be so-and-so, the CAR T didn't work, died of cancer, and there's the number that died of their lymphodepletion regimen.
So, in intuitively, I've always thought of like CAR T in vivo or otherwise as very toxic therapy. But it sounds like their true toxicity is actually the lymphodepletion step, the actual CAR.
No, there's both and.
Okay.
Yeah,
Yeah.
dangerous. The CAR T toxicities are, inflammatory toxicities like,
Cross reactivity.
well, that's actually a good point. So you can get, the cells can have on target but off tumor toxicity, right? If you have an antibody that can cross react. The common toxicities are things like cytokine release syndrome, which is an inflammatory injury that, in its most severe, can cause vasodistributive shock, or something called ICANS or immune cell associated neurotoxicity syndrome, which we don't really understand the pathophysiology of.
Your immune system freaks out. They're all like sort of shorthand for your whole immune system goes like,
Is in vivo CAR T clearly seems to be doing something. Is it as cut and dry as, because in vivo CAR T works, in vivo TCR-T should also, like the tech tree is already completed. You just like repeat the exact same process.
Part of the tech tree.
What's missing?
Okay, first of all, we don't yet know if in vivo CAR T really works.
Okay.
I think that the autoimmune setting is sort of like, it's much easier than cancer. So if you want to just clear your B cells for a week, that's different than you want to actually eradicate every single cell. So that's one limitation. Then the receptor is really different. So like a CAR, it has its own signaling built in. So like how much of the T cell receptor do you need? Do you need to get rid of the other T cell receptor that you're, there's like a lot of like technical little nuance to like, okay, I'm adding a second T cell receptor to all my T cells. Are some of the T cell receptors they already have kind of dangerous for me to be expanding? Or are they going to compete for CD3? So the one I'm adding is not going to work as well. And so there's a bit more to figure out.
Yeah.
So the tech tree is not as built out as you like, but I do think that the fact that in vivo CAR T is having a kind of like bonanza and everyone's going to enter trials this year, that's useful. You learn a lot about like delivery platforms and like how to get the constructs into cells.
Mm-hmm. On the subject of things that are not necessarily cancer vaccines, but they're like strange. I stumbled across this line of research called GVAX, where you take you harvest tumor cells from a patient. Mm-hmm. You genetically engineer them such that they like pump out a particular protein that dendritic cells like really perk up at, and then you infuse them back into the patient. It doesn't seem to work.
Yeah,
but to me that feels like you're throwing the full book at the immune system, saying like this is the tumor, pay attention to it. How come that doesn't seem to do anything?
Well, I think like whole tumor stuff in general has never really worked. It was like a really like nice idea. And there were tumor lysate vaccines of every imaginable flavor. I think this gets into the issue of like think about the information problem you're asking the immune system to solve. Like it was not able to lock onto the signal, when the tumor was alive in the patient's body. And probably that has to do with like how hard it is to sift out what's distinguishing. Like you wanted to learn a very small number of distinctive PMHCs, peptide MHC complexes that, upon, if this peptide MHC complex is presented by a cell, you should kill that cell. There's not that many of them that are distinctive, right? And, 99.9% of the ones on each tumor cell are actually self-antigen that you shouldn't be killing. And you need the immune system to really constantly figure it out and then pump out a huge effector population to do the killing, because they have to like go all over the body and extravasate into every little niche and look for metastases. So, it might immunologically work a tiny bit. Like you might get a little bit of responsiveness to some tumor specific thing. That's sort of the general story with tumor lysate is like get a little whisper of, oh, this looks interesting. Like, yeah, maybe we should be a little more cautious about this peptide MHC. What you don't get is this really confident thing that you get like with a viral infection, where like these 9 amino acids from the spike protein, if any cell's making them, kill them and raise a giant army and make sure that we're all going out doing this job.
Yeah, there's just a ceiling to non-antigen specific interventions for that reason, for sort of the basic information processing reason. And to overcome that, we're going to have to get tightly focused on the very best specific tumor targets.
One of the things that I found very interesting about when it comes time to designing, or like figuring out what neoantigen you want to put into the vaccine is the concept of immunodominance. I would love for you guys to just like talk about that.
It's a big extra challenge that I wish we did not have, because you can have a vaccine that, let's say that you're doing a pretty good job with your selection and half the stuff you put in the vaccine is actually on the tumor. That would be much better than people currently do. But let's say you get that right. And so you encode them all as mRNA, and then you have some mRNA construct with like 20 targets, of which 10 are real and could be the determinants of tumor killing. And then you give the patient the vaccine and they get like a sky high immune response. You use a really good, a spleen targeting IV platform. 20% of T cells recognize a target. It's awesome, but it's just one. And it wasn't one of the ones that are actually on the tumor. And so the vaccine does nothing.
Yeah.
That is sort of what happens right now, especially with the mRNA platforms. But in general, this happens with all vaccines. The immune system has a kind of like multi-tiered competitive dynamics that tries to concentrate the immune response on just a few things, which I think also probably has some sort of evolved information processing role. The immune system will not just passively react to everything you throw at it because it has a budget. There's like a certain number of T cells and it's trying to get a concentrated response. So there's a few places where this happens. One place is that the antigen presenting cells, they take up your vaccine. They might not make every single thing in the vaccine. Then there's also kind of a local neighborhood in a lymph node where, there's this sort of competition for like, oh, this clone is winning, this T cell clone's winning and it has a certain target and it's suppressing the other ones. And there's also like other competitive dynamics that are just in circulation. And probably even more that this is like the ones I could think of right now. And Ben's probably got a bunch more, but like immune cells compete.
Yeah. And from the perspective of the individual T cell bearing a single T cell receptor, that's probably reactive to, at most one of the antigens in the vaccine. It will then compete with all of its brothers and sisters around bearing different TCRs in the setting of general stimulation. So I think this gets at the vaccines were originally designed, the neoantigen vaccines, with the sense of let's give as many epitopes as we can in the vaccine because we know our predictions are imperfect. And so if we just get one or two that are good, that's a win. I think we know now that might not be a win because the ones that you get may prevent the elicitation of better ones.
This is maybe like, there's no known answer to this, but what's the evolutionary point of this? Like, it seems to me that the immune system is almost optimizing, like playing like a lottery game of like, I'm not going to uniformly respond to this threat. I'm going to pick one of the potential responses and just move forwards with that. But that feels obviously worse than starting multifaceted and then slowly like affinity matching your way to the real threat.
So it's a historical coevolution, I think, between, say, pathogens. Because for the most of human history, right, we haven't generally been old enough to have to worry about cancer.
Sure.
So maybe specifically viral pathogens, the MHC locus, and then stochastic T cell receptor generation in T cells.
And I think that there's also first of all, I think antibodies do a better job at breadth than T cells. So if you look at antibody responses, you do get dominant clones, but you also get they get the second chance at diversification. So like somatic hypermutation can sort of take them in more complicated paths. Your T cell compartment's floating around with a fixed diversity. It's like every single T cell is essentially a random classifier and you're just reweighting those classifiers. So I think that if you try to write an algorithm like that, you'd come up with something that's trying to concentrate on a smaller number instead of it's like L1 norm penalized, not L2 norm penalized. And I think that's one thing is that just like to get that kind of learning algorithm to work, you are going to get a little bit of spikiness in which clones get to win. I think also like there are diverse T cell responses to viral infection. If you look, like just whenever you understand the evolutionary rationale, I think looking at viruses is more informative. And you do get T cell responses to, someone gets a COVID infection. Usually there's like they'll get T cell responses to like 2 peptides from the nucleocapsid and 1 from the spike and 1 from the E protein. And so like it's not all one peptide MHC, but there's some effort there to keep it from being like from every possible peptide MHC.
So you can think about immunodominance at the population level or at the individual level, right? The individual level, it's the mix of the pathogen's genome with the individual's TCRs and MAC allele haplotype. And, the possible set of possible pathogen-specific responses is much larger than the actual set that ever obtains in the individual. And then there's the population layer Alex was alluding to, which proteins in the pathogen or protein subunits are more likely across the population to elicit responses, right?
Are there like population level, like for any arbitrary neoantigen MHC pair, is it at all possible for me to design like an ML model to predict like how immunogenic and immunodominant is this going to be?
That was possible.
We have grad students working on it. I think we're not the only ones. Other people have MS companies that sort of claim to do this.
I guess one argument for why it's impossible is that everyone's T cell repertoire is so like different.
Yeah.
That the competitive dynamics are going to be different person to person.
I think there's probably at least like 2 layers here. One is, could you model some intrinsic, properties of T cell targets that are more likely to make them the winners versus what is this sort of like intrinsic stochasticity of that competition? And I think that you probably can attack the like first layer where, there might be like, there's a competitive dynamic going on, but like, really dumb things we know about immunogenicity are that if you have, so when a peptide is in the MHC, its ends are usually buried and the middle sticking out. And if you have bigger residues in the middle, T cells can see that more easily. So it's kind of a like really naive 80s style, like chemico-informatics type feature, but it does roughly hold. And so if you had 2 different peptide MHCs, one of which was like all glycines in the middle, one of which is like really big side chains, then the one with the sort of bigger peptide is more likely to win the competition. It's not guaranteed to, because then there's a whole extra layer of just stochastic competition.
Is there's been a few neoantigen cancer vaccines that have gone through phase 1 and 1 for phase 2 trials. Are they required to share how they've selected these neoantigens from patient to patient, or is it proprietary?
They are required to register with the FDA how it's done. They're not required to publish or share.
And the FDA had like a funny moment where like when we were starting trials on Mount Sinai, like they accepted very high level descriptions of the pipelines. Like I just wrote up some text that was like, we predict mutations. And then I just kind of like wrote up a blog post level summary of everything without too much technical detail. And that went into all the FDA documents. They're fine with that. And then there was a moment where they kind of had accepted enough of these and hired someone who really understood the informatics. And they went back and they asked every single person running any of these trials to fill out these very detailed workbooks about like, what are you doing at every single step here? Which versions? How are you running it? And so they like kind of realized that there's technical details they should care about. And I thought it was interesting because it sort of seemed like at first there wasn't a full appreciation of how much of a computational drug this is. Like they really cared so much about peptide stability. Like we're going to make these peptides. How are you storing them? What's your, how are you validating that they're stable at 6 months for any redosing? Like they cared about the like chemical composition of the vaccine. And it seems like there was like, they just didn't even know how to think about the fact that they're different every time. And then at some moment they're like, wait, this is just like a Python program that picks these drugs for every patient. And then they went back and really wanted to know like what's going into your pipeline.
How secret is this pipeline in a sense of like if some representative from like Moderna and BioNTech was here to like share with you exactly how are they creating this particular like cancer vaccine, like how are they selecting the neoantigens? How useful would that be to you versus you kind of already suspect like this is what they're doing?
We've asked them in various ways, and they don't listen. I mean, broad strokes. Yep, it's very similar. And then the software that comes out of our lab is all open source,
Sure.
so others can look at it and use it if they want.
And I know that
Yeah.
like some of them, I won't get into details. I know that like some of the stuff that I wrote at Mount Sinai that we've kept developing at Pearl, OpenVax, that's pretty open.
Yeah.
And I know that that's been used beyond just Mount Sinai, or at least components of it have been. And then I think that there is this kind of like, there's a desire to say that there's some sort of proprietary, like that we have a real edge because our algorithm's super good. I usually view that quite skeptically unless they really have a good data source that would explain why their algorithm's better than usual. Almost all of them are something that's like a variant caller equivalent in Utrecht and Strelka with the thing that's sort of like NetMHC Pan and McFlurry predicting MHC features. And the times where something surprising comes out is because they're investing heavily in data generation. And then they're modeling some biologic, like the edges in understanding the biology better, not in like, composing existing tools or making machine learning models of existing data better. So you really need to like have an insight in what are we missing about the biology and then model that.
I was expecting you to say like, oh, they probably have a better like peptide MHC binding model. Like, what do you mean by there's like some aspect of biology? Like, well,
Okay, so I'm working on McFlurry 3 right now, right? And this is off like public data, but I've been like combining it sort of in non-obvious ways, like just really thinking about how does antigen processing work in different types of tumor cells and how does it work in the antigen presenting cells so there's a little bit of a mismatch. Like your vaccine goes to antigen presenting cells and then they try to recruit T cells that are going to go kill the tumor. And the way that the vaccine's chopped up might be different than the way that the source protein's chopped up in the tumor. And so like trying to account for those differences, just like thinking through the like mechanistic chain of how do I get a killer nanobot RB to really go after the cancer like at a detailed level. I think if you then realize you have a gap, like, oh, we should do more mass spec of this sort, or we need to mass spec the tumor versus the APCs from the same people. So I think that generating data around wherever you perceive gaps in knowledge potentially adds quite a bit. Whereas there's like a flood of these MHC peptide affinity models. Like 10 of them come out every day. And it's very easy. It's like the most predictable part of biology. And none of them really have any advantage over any others. Like it doesn't matter which one to use because they're not addressing they all work well. And then the rest of the biology is totally uncharacterized.
Go ahead.
Yeah, no, just if you kind of bring down the problem of antigen identification, the first step is finding the tumor specific variants and whether they're expressed. And there are tons of tools to do that. And the tools are actually fairly easy to benchmark, right? You can benchmark DNA and RNA experiments pretty easily. But then after that, you have to figure out of the RNAs, which ones actually get translated into protein. And then how does the protein get processed? And that is very, very hard to actually benchmark in any reasonable way. So you don't know really how great the tools are or your tool is. And then after you see that, you have to, the next step is predicting loading onto the MHC, and the MHC getting to the cell surface. And peptide MHC binding prediction is a part of that, but not the whole story. And, you can benchmark that end to end with immunopeptidomics, but you have to really do the expensive version of immunopeptidomics. So people won't do that either. So one of the things our field desperately, desperately needs is true benchmarking so that we can improve our tools, and or know which tools to actually use.
Like benchmarking at like these intermediate steps.
Yes. And the end-to-end process.
And I think that some of the intermediate steps are invisible, so they're hard to benchmark. So like, if you wanted to draw like if you look at, like the vibe coded cancer vaccine stuff, like their understanding of what's going on is like, oh, you put the mutations in a binding affinity predictor that tells you what's on the tumor, right? And like 0.1% of those are going to be on the tumor. So if you wanted to kind of get closer to what is actually on the tumor, you have to start thinking about stuff that's not modeled well and also hard to measure. So how does the proteasome, which is the main chopping up proteins thing in a cell, like how is that chopping up the source protein with a mutation in the tumor? Did you look at the state of the proteasome in the tumor? Like which subunits are being expressed? And in the antigen presenting cells, which may have an immunoproteasome, it's got different subunits. So, okay, now we've sort of figured out how it's been chopped up and some of the pieces line up and some don't. It turned out the tumor is deficient in TAP. So it's not getting into endoplasmic reticulum at all. So you really can only look at proteins that are localized to the ER, and you have to ignore the ones at the cytosol. Or no, it has TAP, but it's missing ERAP2. So some of the trimming as you're loading is not, so like that kind of stuff that really does affect you. We look at experiments of like mass spec of the MHC ligands with these different deficiencies, get totally different sets of peptides. And people like, if they even knew to think about it, usually are kind of overwhelmed and they just go back to like, well, I hope NMHC pad does a good job.
Or, whether tumors are surrounded by sufficient concentrations of interferon gamma changes what they're.
Interferon gamma totally changes all those subunits. So like,
but this is again why I'll make yet another plug for immunopeptidomics, because it doesn't matter if only 1% of your predictions are real if you can use immunopeptidomics to actually find the real ones, right? So if you can use all the predictions to get you into a zone where you can actually make a reference library for immunopeptidomics data analysis, which you need to do, then you use immunopeptidomics to find the real ones. Then it's hard, but then you're actually good.
Yeah. You don't need to understand the intermediate biology.
Yeah. And I guess, Alex and I have had this discussion, but eventually we're going to understand the intermediate biology well enough to make those predictions. But until then, we need to know the truth to work from, to then go back and study that stuff.
Okay. So clearly the process of identifying peptides within the MHC is incredibly complicated. 0.1% of the candidate space is actually there. At the same,
don't quote me on that.
Sure.
Some recording of it, like a small amount. The exact amount varies depending how you do it.
Yeah. Despite it all, though, it seems like there is at least two seemingly success stories of neoantigen vaccines. One by Moderna, one by BioNTech. I think the BioNTech is for pancreatic cancer. Moderna is for melanoma. How what's going on there? How are they able to solve such a seemingly impossible problem?
Right. So, one thing so there's like sort of two different directions to go there. One is to talk about, how close is this to really working? And the other is like, do we currently have the working manifestation? And my hunch is that we do not currently have the working manifestation. So I don't think that either the, BioNTech pancreatic cancer vaccine or the Moderna melanoma vaccine are it, in the sense of they will get clinical responses that you could attribute to the vaccine at a rate high enough that they care about commercially. They might get an approval. It really depends on exactly, how the Moderna phase 3 trial plays out. But I think that what's useful about them is they give us additional hints of success. In my mind, the really strong success cases are not those. They are that we can do TCRTs against cancer testis antigens, and that seems to work pretty well. We, in a smaller subset, can do sort of TIL therapies and TCRs against mutational antigens. Also sometimes works. So like T cells can kill cancer cells based on tumor-specific MHC presented targets. That's definitely true. We also, for the shared targets, have a few success stories, viral ones and CTAs with BioNTech's FixVac, where the vaccine itself actually has a clinical effect separate from any other therapy. You could just point to it and say, the vaccine did that. Small minority of patients or very early stage cancer, but that still works. So you're kind of like, we're at we're in the neighborhood of success. And then you get to the trials that have currently been running, and they do have some hints that they might be working. They also have some sort of complicated other things to figure out. So they're all combinations. There's a lot of agents active. Only the Moderna trials are randomized. The BioNTech one is not randomized. So you have to start peeling apart pretty complicated chains of causality. But I'm going to be optimistic and just say like, okay, we know some things about them. So could we construct a story by which they're working? I think the BioNTech vaccines have maybe 20 targets. And they do seem to, in a subset of patients, get pretty strong immune responses. And those immune responses are concentrated in the patients who are still alive from their 16 patient trial. And I think that it's definitely possible that some of those patients are alive because of the strong immune response lining up with a lucky choice of antigen that then is actually on the tumor. So I don't think that all 20 antigens in the vaccine were on the tumor. I think probably most of them weren't. But for a subset of the patients, you get an alignment of strong immune response and tumor presented mutational target. And that could have clinical benefit that's manifesting in people with a really nasty cancer, not having that cancer now. So I think that feels like a hint of success for which we want stronger evidence. And I think the Moderna story is kind of complicated because they seem to, it's hard to say for sure because I think neither company wants to run big comparisons of their platforms. No one wants to lose. But Moderna seems to have a less immunogenic vaccine. They have to inject a much higher dose, so they don't seem to get the same kind of big circulating T cell responses. On the other hand, they have more antigens. They have 34 antigens. So they might be picking out more targets that are actually on the tumor. But
that also means they have like more opportunity to lose, right? Because of
immunodominance. Yeah. And I think that this is a place where like looking at they don't often show as much immune monitoring as you'd want to see. And it's not comparable across platforms. And so immunodominance is not like a total curse here. Like you do sometimes get, in one patient, there's one response, but in other ones, there was 5. And what they mean by response kind of changes quite a bit. So I think that there is a way by which you could imagine the Moderna trial also having clinical benefit for a subset of patients. I think to me, it's slightly less clear than the BioNTech one because BioNTech has this kind of necessary precondition for success, which is these huge T cell expansions.
Sure. Yeah.
And so I think that given that background of we know that a bunch of other T cell therapies and vaccines have had clinical benefit, and here's a mutation targeting vaccine that gets strong T cell responses, it doesn't seem crazy to me to say that some of those patients are benefiting. But it's probably not all 8 that, that are still disease free at this point.
Yeah.
And I don't know exactly how many. It could be 1, it could be 3. And so I think that the job now is to take everything we know and all the successes we've observed and then start maxing out these like orthogonal categories by which you need the vaccine to succeed in multiple ways. And we know that it can sometimes. So now you just need to start dialing it all to 11.
I guess I'd like to hear from your clinical experience, Ben. Resected, like for me, PDAC just feels like, okay, you get this, you die. And maybe the patients who are able to get resected, it's early stage enough, such that's fine. But does it almost always come back? And the 7 out of 8 patients who, 8 patients had immune response, 7 of those patients never got recurrence. Does that sometimes happen, or is it extraordinary enough that you feel?
Yeah, with all caveats that I'm a cell therapy clinician and not a solid tumor oncologist, much less a pancreatic cancer doctor. Yeah, I think it can happen.
Okay.
That say people have a treatment course that doesn't include the vaccine, they could still be living without pancreatic cancer if they had complete resections.
Mm-hmm. And I was trying to be sort of optimistic in that I do think this stuff is starting to work and that we're in the neighborhood of making mutation targeting vaccines that work. On the other hand, it's a 16 patient trial.
Sure.
The 8 patients with stronger immune responses have many favorable baseline characteristics in terms of where in their pancreas their tumors were, the degree of lymph node involvement, the size of the tumor, other factors. If you just go through that supplemental table of all the characteristics of the patients, every time you look at something that could have a difference in their outcome, it usually is concentrated in the patients that were in the 8 with immune responses who now are sort of perceived as having been, clinically received benefit from this vaccine.
Yeah.
So there's a lot of confounding there and a very small sample size. So I don't think this is the trial in which you could point to and say, oh yeah, we got neoantigen vaccines working. Look at the, pancreatic cancer trial. I think that it's sort of like when you line it up with the other evidence, it feels like you can't totally dismiss it, which is better than the entire past generation of neoantigen vaccines.
One thing I was trying to like tease out with the help of Claude, and I wasn't really getting anywhere, is pancreatic cancer and melanoma are on 2 different sides of the spectrum with regards to tumor mutational burden. Should that like weigh into this at all when you're deciding how well neoantigens actually work in this type of cancer, or is it kind of irrelevant to the problem?
I think if you have more predicted neoantigens to choose from and features to prioritize with,
Mhm.
maybe you're more likely to have a good one that gets into your vaccine.
Sure.
Because it's actually there and it's actually presented. So it does seem like there's a higher prior for melanoma to respond over pancreatic cancer.
And I think like my experience in doing some of the vaccine designs, like a couple different trials, is that when you have a high mutational load, then everything at the top looks great. Like you're like, oh, there's like 700 RNA reads spanning this mutation, and the predicted affinity is like single digit nanomolar, and everything just sort of really lines up. The clonal fraction of this mutation is 100%. Like they all kind of look like that. Like smoker's bladder cancer looks like that. Melanoma often looks like that. When you go into low mutational burden cancers, you're really scraping the bottom. We're trying to fill 10 peptide vaccines for glioblastoma. And when you get to like, peptide number 8, you're really like, oh, should we lower the minimum number of reads? Like you start twiddling with the filters to include everything. Cause like, one of them had 12 RNA reads and the other one's got 3. And so like, you just definitely end up with worse candidates in the vaccine. And I think that pancreatic cancer is not going to be as bad as glioblastoma, but it's closer to that, where you're not able to prioritize things that all look really promising to you. You have to kind of, at least set your thresholds kind of low in order to fill out your vaccines.
Because I feel like the BioNTech one, I think it failed in CRC and smooth muscle. So like I forget exactly what type of muscle cancer, but like I don't know, does that update you one direction or another?
And I thought they also had a melanoma one, right?
They also had a melanoma one that failed, but it was not resection, I think.
Yeah.
I think so.
I have been, until really recently, kind of a neoantigen pessimist. Like we ran all these trials at Mount Sinai, and then I was like, oh wait, like these things don't work. We should either fix them or find a different therapy. Ben and I, through kind of a research journey, found cell therapy, and we're like, oh, we should really focus on cell therapy for well-characterized antigens. You should always do mass spec because the predictions are useless. And really recently, I've kind of realized that like, oh, you actually probably could get the machine learning good enough. Like we do have a lot of new data sources now. You can think through the biology better. I'm feeling optimistic that like some of the stuff we're building is going to help make the in silico predictions possibly good enough, combined with the fact that BioNTech figured out a great vaccine platform. And there's a couple other ones, like maybe the Elicio vaccine's good. It seemed like Gritstone had an okay one. So the platform question is also a little bit more settled in that there's vaccine platforms that look decent. So I think that if we make the informatics better, make the vaccine platform good, all this stuff could work. My hunch is that the trials that we're starting kind of at the dawn of this era, which like the designs are locked down, like they've, once they have the platform submitted, they don't want to change it. And so the sort of design decisions here informatically are probably pretty primitive. I don't think I might be wrong. Some BioNTech might call you up and be like, this is total bullshit. We did like $20 million in mass spec to make a better model. But, and I know they bought Neon, I think, because like Neon was a vaccine company that had a lot of mass spec data. Mm-hmm. So like, I'm sure that is on someone's mind there. I don't think that these trials have really integrated the advances in mass spec profiling and data generation or in deep learning. And so they probably are mostly putting noise into the vaccines.
If I'm thinking, if I was like a rational pharma company, what it would probably do is like set up a pipeline of like cryopreserved frozen tissue to mass spec and train a really good model for what is actually surface on the surface of a tumor cell, given that like this is its genome. Do you not think that's what they've done? And they've actually just like to pick the naive, like maybe open vaccine looking approaches.
Yeah, I think that the stuff that they were starting before all that data was generated was kind of naive, the way that we built for OpenVax and the way that lots of other trials have run. And but at the same era, there were people and really like two companies kind of like bet much of their strategy around like doing what you're saying, which is Gritstone Oncology, who I think now doesn't exist, and Neon, who got sold to BioNTech like quite cheaply. But their idea was that they would generate a bunch of mass spec data and then figure out what's really on the cell surface. It was more nuanced and technical than you're saying. They weren't all different tumors. They weren't looking for mutations. They weren't doing the weird fancy kind of mass spec that Ben and I are talking about. So they had to like calibrate more off of self antigens. But they did generate a bunch of data and they did build models. And I worked with a sponsor research agreement with Neon to build machine learning models on top of that data. And I think that probably was, moving them in a better direction as far as the quality of the predictions. However, like you need all the things to work.
Yeah.
So I think that, my sense is that Neon made some progress in the predictions using mass spec data, but they're using a really weak vaccine. And, Gritstone did some stuff in mass spec, but I never worked with them. I don't know like how it ended up. And their vaccine was better than Neon's, but still, I don't think they just didn't hit the whole checklist of stuff.
I think Gritstone was not doing personalized.
No, they had one.
They did.
They had a shared that are personalized, the Granite and Slate, and they both failed. And they went bankrupt.
Good name. So
Yeah.
the if I was interviewed on the street and someone asked me, is a cancer vaccine possible? I would probably say no, because there's like within a solid tumor, there consists so many heterogeneous subpopulations of tumors, and like, each one of them is going to have a completely different MHC presentation. Why? Like I know for like pancreatic cancer, they're very like it's a very like clonal cancer. Like many of them they're going to share near antigens. But I would expect for something like melanoma, it would be like highly heterogeneous. How come it works?
Yeah. So now you're getting another design point, beyond my soapbox about immunopeptidomics. I also have a soapbox about you should cover all the tumor subclones with your vaccine epitopes, right? And you can measure that if you have a sample and you can do single cell RNA sequencing and you've identified the antigens and you know what their coding transcripts are.
But you're going to measure them for the sample that you picked.
That's right. So yeah. We can't see all of it.
And on that somewhere else is going to be kind of different.
Yeah. So then let's like overlay that with what's the right clinical context for a vaccine. It's probably not huge burden metastatic disease, but these vaccines that seem to be working better are given in the adjuvant setting after surgical resections.
I just think when you have, I strongly think, I think that's become a meme that like the whole vaccine industry drafts off of. And I think that actually when vaccines work, they'll work even with established diverse disease because your targets are great. Because I think that like, and it's literally that one patient in the BioNTech melanoma FixVac trial gives me hope. But I think that like, if you have a good target, you chose well so that it spans clonal diversity. It's like they all still have to make it for some reason. And your vaccine's really good and it's really presented. I think you can get what.
Don't you think you need multiple targets then?
Yeah, I think they'll escape from the one target.
Yeah. Like each of the neoantigens you put in the vaccine is like a good one. And so you cover the full breadth of all possible.
Yeah.
And I guess like, so like you're in your hyper optimistic case, you think a patient with like very metastatic stage 4 cancer in the cancer vaccine of like the 2030s, it should work in that case.
I think it's not crazy to imagine that. I think there's a few things that work in your benefit. So let's say that you get, you have a great platform and some like, large fraction of your immune system is now mobilized against the cancer. So you all over the body, you're getting these encounters with T cells, and they're cytotoxically hunting down the tumor cells. And then hopefully, and this is a hopeful thing, but you will then reveal more of the tumor antigen to the immune system. So you get some antigen spreading all over the body. So different draining lymph nodes of these encounters are then eliciting additional anti-tumor immune responses. I think that's a way by which you can go from like 20% of your T cells are going around killing cancer. They alone are not covering the full diversity of the presented tumor antigen. And that like vaccine-induced burst of T cells, that's like 20% is your peak. They're declining. So like that is not sufficient on its own to do it. But coming up behind them, you have additional waves of T cells that are being primed from the killing that first wave is doing. And they're also going to cover more of the diversity. But I think you need a big burst. Like it can't be like a subtle, like a little bit of immunogenicity doesn't do that. You need a really big burst of cytotoxicity.
I did see people like poking at the possibility of like you'll get these like polyclonal responses. Like where does that come from? Like my impression is like you give the T cell all the information that you know about the tumor, it goes off and performs the killing of those particular like tumors expressing those neoantigens, but then it stops after that because it just doesn't realize that all these other things are also worth killing. Like, where do you get this extra diversity from?
Like, why do you get the spreading of the? So, I mean, I think that it doesn't always happen. I think that like to figure out the context, like what makes that happen most efficiently, it's important. But the basic idea is that when a T cell kills a tumor cell, all that debris with the sort of cytokine context of the killing goes to the nearest lymph node. So now you have a bunch of antigen presenting cells that are like, oh wait, this was T cells were killing cells that were making this.
Yeah.
And this is just all the debris of the cancer.
But don't you go back to the previous problem of like most of this debris is cells?
Yeah, it's hard. It's 100.
There's some adjuvant effect from whatever you give with the vaccine as well. So maybe your dendritic cells there.
But I think it's like always the immune system has this problem, and it's not like it's always succeeding or always failing.
Sure.
The context matters a lot. Like the debris draining in the context of a bunch of T cells killed it that are secreting all the cytokines and sort of, like just all the general signaling of a bunch of cytotoxic T cells showed up here and killed like 10 million cells. That pushes this imperfect system further towards like, okay, figure it out, find the thing. Something here is dangerous. We need to elicit more responses.
I could buy it. I mean, I hope that the future you're describing comes to pass. It feels like I don't know. I when I read these like these clinical trial readouts, I got initially very excited, and I saw like, oh, this is resection. So like in an ideal case, you are able to do this in the metastatic.
So where they're positioning for is just an easier problem, and it probably makes sense if you don't yet know like the magnitude of impact you can have, but it also makes it much harder to know when you're like having any impact at all.
Yeah. The clinical trials have to be bigger and run longer so that you can see differences in groups and recurrence. But if you have widely metastatic disease and then elicit a CR in 2 months, I mean, that's a case report in the New England Journal of Medicine, right?
So like on one hand, I would expect cancer vaccines not to work because of like there's just so many subpopulations. On the other, like on a similar note, I would expect there to be almost antigenic drift over the months it takes to actually manufacture these things. Is that just practically speaking not really a concern because there's no selective pressure to pull away from these particular neoantigens?
It's definitely a concern. I mean, we've seen that very thing happen in mouse models.
Okay.
So we presume that it happens in humans as well. And that's one of the limitations of the earlier approach of trying to find antigens by just finding antigen-specific T cells without like doing immunopeptidomics at the time you're going to treat. What you're seeing then is just an immunological snapshot of what the T cell populations have seen of the tumor in the past. They exist because they were elicited before. That doesn't necessarily mean that the tumor clones and subclones that are there now are still expressing and presenting the targets of the T cells that are there trying to fight them. It's evidence that there may be some antigen exposure of the T cells at some point in the past. You don't know how current that is.
Yeah.
So I think, again, back to, immunopeptidomics as soon as possible before treating I think is how you mitigate that as best we can now.
But I think that like your point of it's a dynamic system.
Mhm.
And so you're never like your snapshot at time of surgery, even if like it does sort of push you to shorten the timelines. Like if it takes 5 months, it's worse than if it takes 1 month.
Yeah.
On the other hand, it's like maybe just a fundamental problem. Like yet another, we get everything else figured out. Then you have to figure out this like additional problem of it's really like a diverse moving target. So like there's an interaction between that drift you're talking about and just the baseline diversity.
Yeah.
Like you're not going to cover the entire cancer and like this one subclone's increasing over time. So you generally have to have something that's like a story for why you're going to get coverage over lots of subclones or like everything you're targeting is just somehow mechanistically linked to it being a cancer, which there's a few cases where you can have that. Like if you're targeting the driver variant, it's hard for the cancer to lose the driver variant.
Sure.
So it makes, KRAS and P53 mutations really appealing, but it's not useful for most people. They're like not presented on most MHCs. And so then you have to figure out what else is there that's like still closer to linked mechanistically. But then for all your passenger-y things, you do want to have some sense of like diversity of coverage and also like some mechanism by which you hope other antigens get picked up. You also could do this adaptively. So you could like imagine the 2030s version. It's not going to be a single surgery snapshot followed by a 4-month manufacturing process so that you just hope that it works, right? Like probably it's going to be like endlessly, continuously adjusted.
Yes.
You'd pick up any like cell-free DNA with a hint of a mutation, it kind of enters the set of candidates. If you pick up there's like cell-free RNA sometimes, like if you pick that up and it's got a hint of expressing a mutation, that goes into your like log of here's what I think may be in the body now. Maybe you have other kind of like minimally invasive ways of keeping tabs on the cancer. Extracellular vesicles can sometimes come off the cancer. You look at their MHCs. So I think that kind of like continuous monitoring and then adjustment, and and if it's a personalized therapeutic, adjusting it to be new is not that hard. This is much harder with like fixed composition of matter drugs. Like, oh, the inhibitor for that receptor stopped working. Like, which other small molecule things exist? Like, you're limited by someone having spent 20 years making a compound for you. But if you're making the drugs from scratch anyways, then that adjustment could be much more dynamic.
Has any of the liquid biopsy companies explored this sort of stuff? Like, does cell-free DNA actually point you?
There's definitely interest. I don't know which of those interests I can talk about, but like they definitely are interested in this because it's a very natural use case for their product. Like, if you had a therapeutic that you had to keep adjusting based on a cell-free, tumor DNA assay, then it's kind of a natural match for them. I think they want to be in that business.
And one of the technical challenges is you really need whole genome sequencing for that to work, right? You don't want to be limited towards some set of mutations that you've observed in the initial tumor biopsy and only see those forever into the future. You want to see those changing, but you also want to see new ones arise, the capacity to see new ones arise if they do.
That makes sense. This is a side question, but something that I just recently thought about. Why even do cell therapy at all for like TCR-T? Why not instead just infuse in polyclonal antibodies that are like geared to hit the specific neoantigen?
Yeah,
I could make them.
Yeah, so that's the thing. This is like so there are there's like two things sort of like that. So one is that there are TCR T-cell engagers, and so the way that they work is you have an affinity matured T-cell receptor that recognizes that peptide MHC target, and it's a bispecific, and the other side is an anti-CD3 Fab or something like that. So it grabs any passing T-cell and kind of pulls it in and gets it, to start signaling. So any T-cell is converted to a tumor killer.
Mm-hmm.
It's a really nice idea. In the lab, we have not found them to be as effective as a cell therapy. The T-cell receptor signaling in a cell seems much stronger than what's sort of induced by something grabbing onto CD3 and forcing proximity. There's also TCR mimetic bispecifics, and those are just straight up antibodies. They just make antibodies against the PMHC, and they do the same anti-CD3 trick to induce cytotoxicity.
So it's the making of peptide MHC antibodies that are highly specific for the peptide MHC and not cross-reactive. That's the real challenge there. But if you could say make those at will, then sure, you would want to give them. And you could give them as, antibody drug conjugates if you wanted to, or, whatever modality.
Radioligand therapies,
that would be fantastic if one could make those things.
And I think we're like entering, we're not there yet, but we're like entering a world in which you could theoretically make antibodies on demand for PMHCs.
And that's what I was going to ask you. How real is that field?
And people are doing that. 100% people are doing it. People are definitely doing that.
Okay.
There are challenges specifically with peptide MHC targets. So it's much easier to do this when you have like either a really predictable structure or a true crystal structure from like a protein that's not such a delicate little target area. The peptide MHC complex is weird. So, there's very few of them that are in PDB, and the AlphaFold prediction of that structure gets a lot of the side chains wrong. Like, because you're targeting something tiny. It's like a 9 amino acid peptide, and with like 3 amino acids are kind of buried in the binding groove, and you're trying to recognize the pattern of the side chain sticking out of the groove. And it has some flexibility, so it moves around a little bit. And then if you run those through like AlphaFold or bolts or something, they don't really come out looking quite the way they do when you know the structure. And I think that makes it harder to get something that's like a really specific binder for just residue 4. There's like a valine to arginine mutation in residue 4 sticking out of the binding groove. And the way that it's going to be recognized by a TCR or like a high quality TCR mimetic, it's hard to just de novo design those binders right now. I don't know to what degree that's like a data challenge or machine learning challenge or whatever, but it's like at the frontier of like, what can you do with like Bindcraft or whatever? Those are pretty hard to make.
Yeah, the other piece of it, and this is super exciting, by the way, so thank you for bringing it up. But the other question is, how are you actually going to translate this, presuming you could make your binders well, in a way that the FDA is going to allow you to give it to a person without some type of extremely detailed toxicity screen? Like, how are you going to prove that it's not just specific for the peptide MHC that you're looking for, but that it doesn't cross-react against some other important protein in the body that it would be exposed to such that it could cause toxicity?
Well, I would imagine, I would hope they wouldn't ask questions here if they're not going to ask questions for the neoantigen vaccines or the TCR.
So
TCRTs.
I'm talking about like binders that are, generated, completely novel.
Okay.
Like de novo binders.
So I think that maybe we should come back to this, because I do think that like soluble therapeutics that's an interesting direction to go in. But maybe one thing we haven't talked about that's like the most important thing about neoantigen vaccines is that they're just this like complete departure from how the FDA has ever interacted with any therapeutic ever. They're just categorically different from all other medicine. And I think that is like really important, because they are a computational process that designs a therapeutic from scratch, and then you make it, for the first time ever, and then you inject it into a person after like fairly minimal testing to make sure that you made what you thought you made and that it's not like contaminated with something, right? But all of the usual logic about drug development is like totally out of view in this process. And I think that is like much more AI shaped than anything else in medical development. So when I see, there's all this kind of like interest in making better AlphaFold type models like ISO-DDE, and this is going to like rapidly accelerate medical progress. I'm like kind of skeptical, because every single compound that comes out of that still enters this really, really slow process. And so like maybe better things enter the pipeline, hopefully, but like they'll still take 10 to 20 years and a few $1 billion to develop. Whereas like, I don't know, our scrappy little trials of Mount Sinai treated 60-ish patients with like hundreds of different compounds that we made totally computationally. Like if you treat every single peptide we put in a vaccine as like a different therapeutic, we're like outcompeting GSK. And like, I think that if you can start fitting more therapeutics into that shape where your safety prior is really strong, so you have a reason to believe, and this is kind of the issue with like, can you do PMHC binding bispecifics, radioligand therapies or something? I think that like the safety logic's not there. So it can't be kind of neoantigen vaccine shaped to the FDA because they'll ask some really reasonable questions about like, won't this bind to some unexpected protein and then just like melt people's eyes? And you won't be able to answer, no, of course not, because you don't know. So I think that the thing that you need is you need this sort of strong safety priors that have gone into fully personalized therapeutics, which are essentially neoantigen vaccines and also neoantigen targeting TCRT, which one company really did.
Maybe the N of one gene therapies are developing along that.
They're in that direction, but if you look at the amount of work they do for that single patient, it's much more than for a personalized vaccine. It's still
like baby KJ.
Yeah, baby KJ. Like the baby KJ is like dozens of people hustling for months to put together this package that like,
But like now that they've done it for one patient, does that not make it easier to do for every other patient, or is it like the same slog over and over again?
I think it can improve it. And there's another example that I like a lot, which is the antisense oligotherapies that are sort of similar in shape to the baby KJ case. So there's what's the name of the nonprofit? They have a podcast. You're
talking with the phage folks now?
No, the phage folks are also a great example of this kind of thing, but they haven't done as much. There's a nonprofit that does antisense oligotherapies for germline defects, and their development timeline's also kind of long. Like it takes them, I don't know, maybe like a year or something to make the therapeutic, but it is for a different mutation every time.
Okay.
And so I think that you have to find those examples and then try to stitch together what makes it safe and fast for them to do those one-offs. But the N of one therapeutics are still like, in terms of like speed and testing burden, like a tier above, like a tier worse than neoantigen therapeutics. We really have, it's computational to synthesis to administration.
For the neoantigen TCRTs and vaccines, what data package did Moderna and BioNTech need to present to the FDA to be allowed to go through the trial at all?
That's another really interesting point is before COVID and the SARS-CoV-2 vaccines, we didn't really know that RNA vaccines of any kind would be safe, or effective in humans. And, vaccines to that point in cancer had largely been peptide adjuvant vaccines and dendritic cell vaccines. And the RNA vaccines were being developed, but
That's fact check you.
Yeah.
Both BioNTech and Moderna had their cancer.
No, I know. No. I know that. But that it wasn't like wildly.
Right. You're saying it gave us a thing prior.
Yeah. Yeah.
Like they'd run small RNA vaccine trials.
Yeah. So if you look at, peptide vaccines predated personalization with Clearasin P53, right? And then the dendritic cell vaccine, the first neoantigen vaccine, I think that clinical report was the dendritic cell vaccine in 2015.
Yeah.
And then in 2017, And then in 2017, you had the back-to-back Nature articles with peptide vaccine and RNA vaccine and became the BioNTech vaccine. And so, they were co-developing, but I guess I feel like the zeitgeist in the world was that peptide vaccines have been safe, have tried for many years, and that's the way forward for therapeutic neoantigen vaccines. And then the success of the RNA vaccines has, at least in my view,
totally flipped out.
Not my opinion of it, but just what I think like the general view of the field is.
But I think it matters to the FDA because the FDA now, if you are delivering a therapeutic that has already been of like, at least the platform has been in several billion people, they don't ask as many questions.
Right,
like the data package is not like, how do you know that your SM102 ionizable lipid is safe? They're like, no, okay. Like every human on Earth has now been exposed to this ionizable lipid, so probably it's fine.
Well, they perhaps may not have any questions about the mRNA itself, but they may have plenty of questions about the fact that like each patient is getting a new set of neoantigens. how you know those neoantigens don't cross-react with something else in the body.
Yeah. So the logic, like when we submitted our neoantigen for Mag4 to the FDA, it was like 78-page white paper and a ridiculously long, multi-sheet spreadsheet document that essentially detailed all the selection pieces with parameterizations and tools and so on. So they can see all of the computational logic end to end. But really, I think what they care about for safety and antigen selection is you give them evidence that this is a thing that is expressed and or predicted to be presented or known to be presented by tumor cells that's not also on healthy cells, such that raising T cells against it would be dangerous. Right. So that's the key safety logic is that.
Mhm.
And that safety logic is flawed.
It sounds quite flawed.
Yeah, it's quite flawed. Okay, so I'll tell you two things that make it quite bad. And this is the thing you have to worry about now that we're closer to a regime of these vaccines really working. So you have to revisit. So the original safety logic was just sort of hiding under the cover of the fact that peptide vaccines suck. The immune responses are anemic. You almost never hurt someone. We have a paper we're working on from a glioblastoma vaccine at Mount Sinai, in which we're not sure if one of the patients who died, who didn't have cancer and they died from a reactivity to a vaccine peptide. I think this might be like the only case in which anyone has ever managed to potentially be hurt by a peptide vaccine. 5 other patients are cancer-free like 7 years later. So it's like, cancer trials are rough. But so like that case, I think had like really special characteristics. They were getting regular injections in the neck draining lymph nodes. And so really like we're like trying to max out immune responses in a way that peptide vaccines usually don't. Usually you get injections in your arms and the immune response is actually pretty wimpy. And so it doesn't matter what you say about safety. You can inject anything and I think you'll be fine. I would feel comfortable taking any peptide vaccine with like, an adjuvant that's the ones that have been considered safe. When you get into vaccines that are like 2+ orders of magnitude more immunogenic, you do have to worry about, what is this really going to cross-react with? And if you look at the famous MAGE-Titin cross-reactivity, the amino acid sequence difference between the MAGE epitope and the Titin epitope is pretty big. It is not a 1 amino acid difference. It's a 4 amino acid difference out of 9. Like it's a pretty different peptide. And the sort of safety logic of 1 amino acid is different in this peptide. So of course the immune system would never mistake this peptide for a healthy one is actually kind of weak. Like T cell receptors contact a few hot spots on the peptide. You're hoping that they don't, that in that sort of limited view of the peptide, they don't mix it up, but they might. Like they're now looking at the whole sequence.
Yeah. So this is why the big, thousands of people have been treated with neoantigen vaccines now. Like seeing the safety signal in the large reports will be really helpful. Help us understand, like, is there a risk of these catastrophic tail outcomes? And if so, how frequent are they?
Yeah. And I think the sort of larger BioNTech trials will probably be informative for like, how often does that go wrong?
Sorry, the MAGE cardiotoxicity thing, if I remember correctly, was a TCR one.
Yeah.
Right? Yeah. Instinctively, I'm thinking, if you are using an mRNA construct that is like only eliciting your existing T cell repertoire, you can naturally worry a lot less about off-target effects.
Yes.
Is that clean logic?
Yeah, it's pretty clean, but like a lot of these things, like, like when I came into this as a computer scientist, I treated these things as, as rules. And I think that they work out more as like reweightings.
Okay.
Like there's a lot of naive T cells that you don't want to turn on. Like if you turned a random naive T cell into 10% of your circulating T cells, you're not guaranteed that the thymus did all the stuff you hope it did.
That's just like, I mean, I guess the existence of acquired autoimmune disease as an example.
Right. And like sort of like, all of the immune related adverse events under like anti-CTLA-4 in particular, but checkpoint blockade in general, all those T cells were in your body and they just required some extra mechanism to keep them from trying to eat your colon or whatever they're doing.
Yeah. So the question is, how well does your vaccine discriminate in stimulating only the peptides you want, or T cell responses against only the peptides you want, versus ones that are T cells bearing cross-reactive T cell receptors that weren't problematic when they were low in frequency, but could become problematic, causing autoimmune disease if they were higher in frequency.
That makes
It's
sense.
like, how often does that set of events happen in any individual? And I think we don't know.
Yeah, we don't know. I think this is like to be learned from the newer vaccine trials plus extra research to be done.
The second to last thing I want to talk about would like maybe it'll be the second to fifth thing I ask. You've been referring to like your work at Mount Sinai and also your work
I realized that as I was like, oh, I didn't really like frame this in terms
yeah,
of
and but like now you guys like you've left Mount Sinai, you're now at UNC, and now you guys also have this this consulting company for high net worth individuals for dealing with their cancer care. I would love to just like go through each one of these steps and like talk what you did at Mount Sinai, what you're doing today, and yeah, how you also play into this.
Okay, so first of all, the reason that I talk about the clinical trials work is mostly at Mount Sinai. So there's some other trials and single patient INDs I've worked with or sort of been involved in, but the trials that I really like got to play a foundational role in like writing the software, helping write the protocols, helping run the trials were all at Mount Sinai. This is not entirely of my own desiring, but when we got to UNC, UNC is just a much more centralized institution. Mount Sinai is a bit chaotic. And I think in that chaos, there's degrees of freedom that don't exist in a larger institution or sort of a more, logically assembled one. UNC makes more sense and there's downsides to that. And so one thing that is nice in the chaos of my last job, which is now a long time ago, 6+ years ago, is that Nina Bardawaj got to build out a wonderful operation. And I worked with her that whole time. And she has a vaccine and cell therapy lab. And she took over a big chunk of lab space on the 5th floor of the Hess building and stamped out 3 GMP suites and could just run a ton of trials. She has a weekly meeting. It's wonderful. There's like tons of immunotherapy trials and everyone's like, all the trials have their like, oh, we need more of this or that, or we need to reschedule these patients. We're going to start a new arm with like, we're going to add FLT3 ligand. And, if you have an idea for a trial, you just show up there and figure out how to make it work. I think that is sort of like having your own GMP facility with your own regulatory people, your own research nurses. I haven't figured out how to do that at UNC. I'm sure other people have some way to spin up trials, but like I haven't had luck with that.
Does that exist outside of Mount Sinai?
And there are like productive investigator-initiated trial groups, but it's the kind of thing I hear more about people like going to China for.
I've always heard like IITs are a China phenomenon.
They're not.
a
There's
thing.
IITs all over the US.
That's interesting.
I don't know if there's the same sort of fully integrated operations everywhere, but like IITs happen everywhere. But having a big university that sort of like aligns its mission around we're going to like, have logically planned how we do all this stuff actually makes it a lot harder.
Yeah.
I think Dana-Farber has their program. WashU has their program.
Yeah, all these
There
early
are-
vaccine trials were all IITs at different
Yeah.
institutions.
Do you like? I imagine like your view of these things. These places are positive. Like they are universally a good thing to have versus like maybe the funding going into like single PI just like mass experiments.
Like, say that again.
Like so, like I guess like the counterfactual of like not having this GMP facility is that the money goes elsewhere into like non-human translational research. Is that a
Yeah.
better use of money for cancer vaccines specifically?
I think that trials are really high value. I also think that the cancer vaccine field has a pretty long history of trials that don't tell you a lot.
Okay.
So I don't know how to like balance those two things. I think that like being able to spin up a trial to test an idea is really, really valuable in a way that decades of mouse work won't really resolve a question. I also think that it does require, some incentive structure or like push towards, okay, but what is the question you're answering with this trial? Which cancer vaccines historically have been pretty bad at positioning their trials to really answer any question.
Do you think like IITs in general are not often ran particularly well?
So I think IITs I've seen have been run actually quite well. They're usually run on shoestring budget
Yeah.
relative to
Yeah, that was my
trials.
hesitating no. It's like I think they're run pretty well, but they're not very well funded.
Interesting. But
Yeah.
so I guess then why is the fact that people are not learning that much from this simply a fact like the underlying biology is very complicated? If you're doing small trials, you're just simply not going to learn that much.
Well, every group is incentivized to, test just their thing, whatever their thing is. Is it better informatics for antigen identification? Is it a better vaccine formulation? And so on. Is it this clinical context or that clinical context? And so you run a small trial with a bunch of variables fixed and no comparison group. And, you can have 300 of those and not necessarily know how to put them all together to what is the actual best way to do this thing we're calling therapeutic neoantigen vaccination.
I think this is like a sort of like a funding incentive structure problem where no one, if you make your trial twice as expensive, but it answers a question well, you're not necessarily going to get rewarded for that in any way, but you now have spent twice as much money.
Yeah.
You'll go from $5 million to $10 million. Your paper is not necessarily much more publishable, nor will anyone give you money in the future to do it again. So I think the fact that they're kind of underfunded and there's not there's this notion of the trialist and they just sort of put the thing in the clinic, but it's not integrated with a research question, maybe is part of why this stuff doesn't quite all connect.
Yeah. So to answer your question before, though, I think you also need mouse models because there are too many variables in the therapeutic strategy to test them all in humans. There aren't enough cancer patients to test everything you might want to test, like different formulation benchmarking, prime and boost strategies, combination therapy strategies, TCRTs in the mix before or after vaccination. And in mice, which are not a perfect surrogate for humans, but if we're honest, a ton of relevant human immunology was first worked out in mouse models and then successfully translated to people. Not everything, but a lot of it. And you have the opportunity to explore much more of the combinatorial therapeutic space to get some intuition about what's the best trial I can write, So if I can winnow my space 95% by doing some careful mouse experiments for a year and then have a much better trial on the other end, I think that's better than doing another less informative phase 1 with 12 patients in it. Maybe that's wrong, but I think if we were really working together as a whole field, we'd have this supportive ecosystem of, mirroring clinical trials with deep immunological investigation and mechanistic understanding, feeding, highly directed bets around human clinical trials with comparison groups in them. But that's not where we are as a community.
And I think that there's one thing that maybe misfires in the trial context, which is that trials always are written and conceive of themselves as on the road to approval. So you're supposed to do all your research in mice, which are like okay immune surrogates. And then once you've made your thing, now you're testing safety. And all of the real research happens in that phase 1 trial that's nominally supposed to be just for safety. And then you kind of hide the fact that you're looking for signals that actually tell you about the biology in there. Mm. And then you do your phase 2 trial, which academically funding is really tough, but no one's going to fund it as a phase 3. So really the IATs are all phase 1 trials that are in our kind of ritual of medicine only really supposed to test safety and feasibility. Could you do it? And so there's not a strong reason to instead write the trials as we're going to do a 12 patient trial, but we're going to do 2 different antigen prioritization algorithms within each patient. And we're going to do mass spec on their tumor samples and see while we're making the vaccine, we'll figure out could we find evidence of anything? Or after we vaccinate them, we'll vaccinate them, with antigens from 2 different sources and see which one's got stronger immune responses. Or we'll alternate, we'll do 3 doses of one vaccine and 3 doses of another one. And we'll also do the reverse group and we'll look at immune responses over, people don't want to write trials that way. They're complicated, more expensive. Those would give you really scientifically valuable information. But if you're just supposed to be testing safety for a fixed product that's supposed to eventually get approved, like you just do it and see if you have any adverse events.
I would imagine that IITs are given a bit more flexibility in this regard, as opposed to like pharma companies where they have some sort of like budget they want to put into this program before they just kill it. For academics, I imagine like you like sky is kind of the limit with regard to
That's true, but how are you going to raise $10 million as an academic?
Yeah, like so like the budget's not there to do the interesting trials, and then no one rewards you if you do it. I think that's like, and then in the sort of broader context of trials, they're not thought of as meant to answer the questions that they could.
Yeah.
So you just end up with boring trials.
So you could design them that way as an academic. And I think you could send a grant application to the NIH for doing that. But even the big NIH grants, with the exception of some sort of special huge ones, don't have enough like funding as part of it to actually do the thing. So I think they would support it and be excited about it. If you could do it for a tenth of the cost,
sure.
then they probably would support more of it. But
Is this like, do you imagine this is a problem in China as well? Or like there's some mechanism to help like ensure that like there's like some community-wide effort to ensure that whatever X modality you're exploring, we work on it at, like across labs.
Like all my knowledge of like the Chinese trial landscape is from reading people's like Substacks and stuff. So I feel like I don't have enough knowledge to answer that. My son, I haven't read about super interesting trial designs. They just sort of like hit on, good therapeutic hunches and then test them very directly faster. That's what I know.
Mhm.
But, it's possible that there's also like some long tail of, like what is the state of cancer vaccine research in China? I have no clue.
Was there any attempt at like while you were at Mount Sinai to try and like get people to work towards a common direction together? Or it's just like the incentives are so misaligned that it's hard to convince people to like combine their hypothesis with your hypothesis?
I think that like, what we worked on was sort of in some ways self-contained and had units of funding. So like test out the peptide vaccines with OpenVax. We had some funding from Pisces, and then, if we add this checkpoint blockade drug, then we get a sponsor that like is making that drug. We were doing glioblastoma and we got some money from the Tumor Treating Fields company. And so like, it was really like just this one, it was like the OpenVax group and Nina's vaccine and cell therapy lab, plus wherever we could scrape money for the trial. There was no like kind of bigger effort there. I think now there's a little bit more interest in like, can you start gluing people together and testing this in a more systematic way?
I actually was not aware of this like pipeline of like pharma giving you some money, like multiple pharmas giving you some money to run this clinical trial and like create some sort of hypothesis for the for-profit companies to then like develop further.
It's a huge thing, but that's why there are IITs in the US.
that makes sense.
I think that the more sort of the simpler sounding way to do an investigator-initiated trial is to apply for like a clinical R01, and that happens to some degree. But I think, and Betty, you can correct me because my whole experience is essentially like one hospital network in New York and a little bit of UNC. My understanding is that it's dwarfed by sponsored research. You're gonna run a trial, but it uses this also ran PDL1 agent, and then we're gonna like pay for a lot of the trial.
Gotcha. And so I imagine like the problem with UNC is you don't have this like GMP manufacturing facility. Instead, you don't have clinical trial people. Oh, you do.
But it's centralized.
Okay.
You gotta get on a more centralized plan.
What's like the difference between centralized versus like,
There's a lot of different people across many different compartments of a big bureaucracy that don't have any reason to like feel urgency around the stuff that you want to do. They have other plans. There's initiatives and other programs, and so like there's a lot of magic to everyone being in one room once a week, and like it's all kind of lined up, and the exact same capabilities, even if they're like much more scaled up and in some ways like more logical sounding, if they're diffuse and there's no like everyone's not in the same room together trying to do the same thing, it just doesn't work out.
it slows it down so much, I think, is the real.
it's interesting because it's okay. So it sounds like even though everyone was in the same room at Mount Sinai, there still wasn't the capability to really work together in common research directions because the money was allocated to test one specific thing per group.
I think that so Mount Sinai is like, a good example of being able to start lots of trials, and they often have like an interesting hypothesis. Like if we add FLT3 ligand to this vaccine and we do intratumoral injection, will we see some tumor regressions? You could test that.
Mhm.
But if you wanted to like do something that's a little bit less traditional, like you want to really maximize the scientific knowledge from the phase 1 trial, no one's going to fund it. Like there's no way to like double the cost of the trial to get more, scientifically valuable things other than does this combination of therapeutics, have some strong hint of working together.
Why is there like maybe this is like a public goods problem and sorry, not public goods problem, like tragedy of the commons where everyone wants this knowledge but no one is willing to pony up the money to actually get it. Because it sounds like the knowledge you would get from like a direct search for scientific knowledge would be useful for everyone.
Okay, so I think like to make it more specific, let's just talk about like cancer vaccines as a trial community, like people starting trials, because I don't want to like talk about medicine in general. I feel like I don't know enough to say it. But like in the cancer vaccine field, there have been a lot of like in the 80s and 90s, people were running like TERT vaccines and these sort of TA vaccines. And then they sort of like were like, oh yeah, but CTAs might be a little better. So running CTA vaccines. And they all kind of come out with the same thing, which is that we got some immune responses by some like, somewhat discordant, weak threshold of immunogenicity. And then they run more of them. And then they run more of them and they run more of them. And like, and they're all phase 1 because, but once in a while they hit, like they could show that the, like we get immune responses and they find a company that wants to take it, that thinks they should try to take it to market. Then they ran some phase 3s and they fail hard. Like none of these did anything, right? But the sort of like IIT world was just running a gazillion of these like trials that all are like by design going to get a couple immune responses. So I think the question is like, how do you intercept that behavior and instead make it an optimization process? So rather than everyone steps forward with the exact same information, these vaccines are safe, their manufacturing is feasible, we got immune responses. And I think like there must be 1,000 trials that had that conclusion. Like how could we have instead had a process by which they start with, we know they're not good enough yet, we want to test if this is an improvement. We want to start doing like coordinate ascent in each trial. I think that that's a funder problem.
I feel that feels very focused research organization.
Yeah. So I mean, it's not like we're talking about something that's unrecognized. I think everyone generally would agree that that's a problem. Say we had enough money, that's what we would have done, right? We would have done.
Yeah, like it's not like
optimizations.
Yeah, like it's not. This isn't like secret knowledge that Ben and I stumbled on. I think you ask anyone in the field, they'd be like, "Yeah, but we never knew if poly-IC was actually better than CPG." And then you ask them, "Why didn't you do the trial?" And they're like, "Well, you could do the trial with the CPG sponsor, you could do the NIH trial, but like they want a poly-ICLC because they think it's like generally safe. No one would have funded the comparison, and so we just didn't do it."
Okay, that makes sense. And that was your Mount Sinai days.
Yeah, Mount Sinai days. Yeah.
Now your what was it, Pathfinder?
Yeah. So there's a little bit of a journey here. So I met Ben through Twitter, and we dug up that first Twitter DM, and it was me asking about the trial that Ben was working on starting. So maybe I could let you talk about PandaVac for a second.
Yeah. So PandaVac was what we were working on, and have submitted to the FDA. But this is the idea of the trial would be to adapt the vaccine if the tumor evolves over therapy. It was going to be a trial in squamous tumors of the lung and the head and neck. So we were really excited about it. And we got initial favorable approval of the IND and submitted the informatics and all that. And we've been stuck for a few years on various manufacturing aspects, and probably more than we should go into here. But we haven't treated anyone.
Is it because of the aforementioned like GMP manufacturing problems?
Well, it's too complicated.
We can send you some email chains after this recording, but I think the diffusion of responsibility problem is really acute.
Yeah, I'll take it to myself. Skill issue for me. I'll also say Alex's experience at Mount Sinai was, inspirational. And I think in general, a very good thing, even though they couldn't solve all the problems. But regardless, I think Alex saw that, we had this registered with clinicaltrials.gov and shout to.
Yeah. And the idea was when we first started talking, I was curious about the trial and how they're going to design it. And then, once we realized we were sort of on the same page about like, this is a really promising direction. This is like algorithmic medicine will eventually be the future of how you make medicine. However, none of this shit works right now. So like having both of those thoughts is rare enough that we really like started talking quite a bit. And then eventually kind of came to this idea of like, I should do a job talk around how to improve cancer vaccines. Cause I had thoughts from the Mount Sinai experience where those trials really were at best unclear. I think the glioblastoma trial we ran may have benefited people, but it's unclear if the antigens we put in mattered. And the other 2 trials I was on, I don't know if they benefited anyone. And there's a 4th one. And so like we treated a bunch of people and I didn't feel like I could point to anyone and say like the part that I, owned, the antigen selection had a benefit to this person. And I had thoughts about how we might improve that. And so, in talking with Ben, he was like, oh, you should turn this into a job talk. Like, this is just like a list of things that are a good research program. So I went on the academic job market, which is a thing I'd never had any intention of doing. And then ended up getting a job at UNC. And I think to the shock and horror of the genetics department, just showed up and said, okay, I'm co-running a lab with Ben. Which I think they were kind of in denial about for a while. And eventually we're just
Still are it.
Yeah, maybe. And because they don't like it. They want you to be, they want clear understanding of what each lab's specialty is, which grants they're going for, who owns which grants. Like, it's just weird for them. So we started working together, year and a half or so disruption from COVID. We tried working on COVID vaccines. We made the only bad COVID vaccine. Literally every COVID vaccine worked except for our T-cell vaccine, which elicited very strong immune responses in mice, and they all died when you expose them to COVID. They were the ones that
actively antagonistic.
In fairness to us, in hindsight, when we started, we had no idea that the RNA vaccines would work.
Yeah.
Right. So like everyone else, we
threw our hat in the ring, but everyone else succeeded to some degree. Like it's literally every every viral vector, the inactivated vaccines, everything worked. We made a peptide vaccine using all the sort of computational principles you use in cancer vaccines. And we made a thing that elicits T-cell responses and does not in any way protect mice from dying from COVID. And I was like, this does feel related to like how well the cancer vaccines work. So we started on this program of like, we're going to optimize the vaccine formulation. We've done a lot of mouse work on making more immunogenic vaccines, started making better informatic tools, got into like long read sequencing as an alternative source of antigens. We then got into single cell sequencing. We found that single cell long read is pretty magical. It's become my favorite modality. We started thinking about cell therapies as a sort of, the upper bound on, what can a vaccine get you? It should be like somewhere below a cell therapy. So why don't we make some of the cell therapies directly? And then along the way, we've also started intersecting more with this thing that I think you're curious about, which is the world of like hyper concierge oncology. Yes. And so, there's a few places where I've intersected with this. There's various sort of research programs at Mount Sinai that had like individual sponsors sort of in the interest of developing things that might help someone in their family. I think that's like closer to the more traditional, like name a building after yourself philanthropy, but these were more focused. Like you knew the guy, you knew what the cancer type was, you knew like why they wanted this research done. But that was still pretty distant for me. There's also a vaccine nonprofit, Jamie Leandro Foundation. And so they essentially exist to like facilitate access to cancer vaccines. And so they sort of, it's based on the idea that like, it's a complicated situation right now where you don't know when these work, for who these work, whatever. But there are cancer patients that they're like, and they're past standard of care, and they would like to try a therapy that at the very least is likely to be safe. and there's some chance that they might benefit. And so the Jamie Leandro Foundation has this whole review board for the individual vaccines. So I spent some time sort of working on the review board, just kind of curiosity, because they're doing this thing parallel to what I had done. And I learned a bit about that, and I helped with some vaccine designs. That has some overlap with, there was a program at the Rare Cancer Research Foundation that also had sort of like, there were like various like patient-focused research programs. Like the Rare Cancer Research Foundation deals with a variety of rare cancers, but some of them come with funding because it's the rare cancer of a person that wants their cancer to be better understood. And so I think through that, Ben and I started getting a little bit deeper into the world of, I think now it's kind of called like founder mode oncology.
Yeah.
And so there was a company that then spun out of the RCRF work. So the main person doing that is Willie Hoos, and he then started a company, Pathfinder Oncology, and Ben and I are like minor co-founders in that. So it was really started at a cafe table with me, Ben, and Willie, and we've kept our labs and have we have still like an academic focus, but through working like as consultants for Pathfinder, we kind of nudged the scientific direction. We helped Pathfinder resuscitate this personalized TCRT company, and then sort of reboot it as something that like can deal better with the costs of cell therapy manufacturing. If the people you're manufacturing for can pay the large cost, then it's less insane to do it than if you want to just try to bring it to market. And generally just kind of understood a bit more about, patients who have the resources to try to pay for R&D in a way that normally doesn't happen. Usually you're kind of
Like it directly benefits them.
yeah, like you're at the recipient end of a societal program. Like we collectively fund medical research, and then individual bits of research get boxed up as IP, and then investors try to invest in turning that into medicine, and 20 years later that pops out as a thing that your insurance will reimburse for. If someone doesn't have any options from that entire pipeline, but they do happen to have $1 billion, it's kind of surprising to me this hasn't happened more in the past, but like now the thought is increasingly occurring to patients that like, if I have my options, maybe I can pay for some new ones.
Yeah. How, if I'm a billionaire with a rare cancer, and like maybe I exhaust standard of care and I look towards these like personalized health concierge services for oncology, how good is it really? Like how much better is it than just like staying on standard of care and seeing how long I can get there?
Yeah, it's a tough question because it's a really heterogeneous mix of situations. I think Sid is a good index situation because he's so public about this. So you can talk to him, you can talk about him, by name. And he has osteosarc.com with all of his all of his scans and genomic data and all that. So this is someone where like privacy is not a concern. He wants the situation to be understood. And I think you could see the possibilities and the limits of this approach from the like really complicated journey that he went on. So he does not have good standard of care options. There's, I think that, I don't know if you agree with this, but like, you stay on the like NCCN guideline course, like I don't think his sarcoma would go away.
Yeah, and eventually would occur, and more aggressively, and more tumors, more places.
But he's tried a lot of stuff, and I think most of it, it's really hard to say whether it did anything. And then there's a few things that might have done quite a bit, but it was through a pretty elaborate hedged strategy that required, I think that employed the services of like all three of the concierge oncology companies, one of which he started, as well as a lot of personal effort and people that he's hired just personally. So I think that like that kind of, what he's been able to do so far points at the possibility of a better outcome than you would normally get at tremendous financial and time cost. Like you need to like fly all over the world and hire a ton of people and, try a lot of stuff, some of which may be somewhat harmful, some of which may be neutral. And, that's like, and that just becomes like it's a huge endeavor. I think most patients don't, even if they could figure out how to do that, don't want to do that. So I think that like that could also be seen as a kind of upper bound to bracket the amount of benefit you could get from, if you're a billionaire, employing the services of someone to try to recreate the like, personalized therapy flow.
I've never gotten like an oncologist take on his like actual osteosarcoma journey. He did have a cancer vaccine arc. He also had like an experimental like FAP drug arc, and maybe a bunch of other things in between. Is it clear like could you attribute causality, or like you could attribute like most of the variance of the situation here is like this like final thing, or it was the cancer vaccine or something else?
I don't think you can attribute causality. I think this is actually, and this is really an interesting point to think about in general, is how much and what exactly can we learn from N of one experiences?
Sure.
Right? Because there's if you think about evidence-based medicine generally, randomized control trials are the king. We want to be informed by them whenever we can. But at the limit where every cancer is a rare cancer and everything is an N of one, it's impossible to do a randomized control trial for every drug or every combination that you might consider, right? It's mathematically impossible. There aren't enough patients, there's not enough time, and there aren't enough drugs that you would want. But even if there were. So you have to think, what can I learn from an N of one context? And if you look at how an N of one trial would actually be designed, it would be for some condition where you could randomize yourself to receive different interventions and with washout periods in between, and see how much better you get from each one, right? And that's actually better knowledge for you than randomized control trial knowledge. Let's say you have a chronic condition, like you have chronic headaches, and you want to figure out which of 5 medicines are best for your headaches. So you randomize yourself to take them for 2 weeks, right, at a time with 2-week washouts, and you keep careful logs of frequency and intensity of your headaches. And at the end of that, if you do it right, you gain some information about which medicine works best for you for headaches, right? In the cancer context, there's a path dependency to everything that happens one after another. So you can't really stop and wash out after individual drugs, because the cancer is continuing to evolve, as your therapeutic strategy is changing over time. So that option isn't open to you. But on the other side, and advanced, say, stage 4 metastatic disease that exhausted all lines of standard of care, the likelihood that a person would get a deep response that lasts at all, or that much less that lasts for some period of time, by doing nothing at all, is minuscule. So that, maybe it's not zero, right? Maybe it's 1 in a million that you could get a deep response by doing nothing. So if you've taken some action that's taken you from, no response to response, and the likelihood of having a response without taking any action is close to zero, then I think you could argue that you have learned something about you and something you responded to. But what you can't do is you can't parse if you've taken 7 interventions in an intermediate span, you can't look back and say, oh, I know it was this one or the combination of that 2 or those 3 that did the job, right? Yeah. So it's one thing, I think we have to think carefully how to learn from these N of one experiences in terms of what monitoring to do, cell-free DNA and RNA and imaging and other clinical monitoring. Can we set these things up such that we learn the absolute most from them? And if more people are doing this, right, if there's not one founder mode on cancer, but there's 100 founder modes or 1,000 founder modes on cancer, then how do we learn at a population level from those 100 to 1,000 experiences such that we learn more and more over time? I think the way it was best put to me in thinking about, why do this at all, is the Mark Labs, who is the founder of Rare Cancer Research Foundation, and himself a cancer patient. He said, developing curative therapies for really hard cancers, we don't know how to do it, and it's happening too slowly. And it's not happening for all the possible cancers out there at equal rates, right? Like some are studied more than others because of incidence numbers and so on. So he said, we have to let wealthy individuals who are both deeply savvy and informed and risk tolerant, the risk capital and the risk to their own bodies to teach humanity how to cure otherwise incurable cancers. And I remember meeting with him some years ago, and that inspired me, what he said. I think it rang true then, and it still rings true now, that if we can learn somehow from Sid's story how to do this better, and others like him how to do this better, and then bring that, once we know how to do it, scale it and make it cheaper and deploy it more widely, that will have been a great thing to have done.
Some of these personalized oncology companies seem like they've been around for, like, at minimum a decade. I think some are even like since the early 2000s. Have they ended up like bringing a lot of knowledge to oncologists that otherwise would have just like never been explored?
Which
Not-
ones are you thinking of?
Yeah.
Like
There's
what
like this-
category company?
There's like this one, I think it's like called like personalized cancer care.
No, I don't know. That is.
It has a very old fashioned website.
Mm.
I just know that they have been around at least like longer than like Sid, I think, was like the most maximalist, but there have been others before.
Yeah, I mean, I think that one, like, as an industry, we'll try to tailor a cancer therapy to you, feels like it couldn't have meaningfully existed until pretty recently.
Okay.
Yeah.
So I don't know what they did, but I don't know what they could have done. Because the options,
Oh, I think you're the one who told me like they convene like tumor boards, and that's like all they.
oh yeah,
Yeah.
that's a separate thing.
Okay.
So
Yeah,
So
tumor panels.
the
Yeah.
the janky website
Yeah.
company, yeah, they're great though. They
They're awesome.
Yeah, they put together really like well constructed informed tumor panels. But in this kind of like what are the degrees of freedom available to the oncologists on those tumor panels? Like often pretty limited.
Yeah,
And like
yeah.
what molecular profiling could they go off of? Also pretty limited. It's really like quite different now versus 10 years ago, because I saw some of these. There were like 3 different programs called the Personalized Cancer somethings at Mount Sinai. And like really didn't get anything done. They all had some version of, we'll look at your genome sequencing and then we'll tell you to take, I don't know, ivermectin or something. Like drug repurposing and, drug sensitivity prediction, things like that. They were just useless. And, they might still be useless. I don't know. But they definitely were useless when the drugs were bad. You didn't have really precisely targeted drugs. There's something like really quite different now in that the molecular profiling is a bit more sophisticated, but then the armament of drugs is getting way better really, really quickly. So, you went from having mostly kind of like dirty small molecule type drugs that, you're hoping you could pick up on some statistical signal of how they're well matched to this cancer, to, well, we look at the RNA, the single cell RNA sequencing, and here's the tumor cluster. It's making, 200 times more of this receptor than any other cell population. And when we look in our catalog of possible drugs, there is an antibody drug conjugate for that receptor, and also radioligand therapy, and also a T cell engager. So we got to pick which one of those 3 we want to use. So I think that is a really different situation that has previously existed.
And I guess another thing is like there's this whole like Rosie's dog thing about designing a cancer vaccine, and I think like one can go back and forth as to whether the vaccine did anything versus like dog eat Keytruda. But it did seem to me like that world could not have existed without access to LLMs and like kind of like teach people like all these things. I'm kind of curious to get your perspective, like your patients. Like, do you find that they're opting for like stranger treatment because like they're operating off of like this like the 2026 literature?
No, and I think the difference is, like, I'll give you an example of what an intervention might be. Let's say that you have a tumor type, metastatic disease, treatable by chemotherapy maybe, but it doesn't usually get, say, HER2 testing.
Sure.
Right? But some molecular studies get done on a broad panel and it's lights up for HER2. Well, now there are multiple clinical products that can target HER2. So then, that person can go to their treating oncologist and say, I know I have cancer X that doesn't usually get treated with HER2 therapy, but look, I have this study that is showing that, my tumor is positive for HER2 and that, that can be done in a pretty clean way, right? You could have like an RUO screening study that then surfaces possibilities. And then sample goes to clinical lab for a CLIA test. So you're still working on your, approved pathological test that can guide therapy. And then the oncologist and the patient can, share decision making together, figure out if they want to do that or not, right?
Mm.
But the product is actually more information for the person.
Well, I guess that's what I'm talking about. Do you find that, maybe not every oncologist is able to stay on top of the literature as perhaps now the LLM is able to. And do you find that do you imagine are patients able to better advocate for their own care in this post LLM world in oncology?
I think they are now, and more will be very, very soon for that exact reason. But you still got to make the testing available, and then it gets back to who's going to pay for that.
That's
right, because now there's a ton of second and third order effects of gating all medical diagnostics and care decisions by manifests of what the insurance company will support.
Mm-hmm.
And one of those is that not everything is widely available, and that's we have to work within that system of constraints, right?
Yeah. I do think that there's gonna eventually have to be a moment in which testing gets broader, and because I do think that pharma's actually done a really good job. I was surprised at the rate at which useful seeming drugs are all coming online at the same time, and that's despite all the flaws people talk about, how clinical trials are hard to start and it's really expensive, whatever. It's still have a ton of new drugs. A bunch of those are probably going to be therapeutically beneficial. And to know if they're useful for you, you would, as a cancer patient, want to get at the very least RNA sequencing, which is surprisingly hard to get, right?
Yeah, DNA, RNA sequencing, and a basic proteomics panel on your tissue slides.
Yeah. But even the Tempus RNA-seq is kind of seen as exotic,
Really?
Yeah,
not everywhere.
Depends on the hospital, depends on the indication.
Yeah.
Some indications, they always get Tempus exome and RNA, but often if you're doing genomics, you'll get an in-house panel that the hospital can be reimbursed for. That's a targeted panel of 500 genes. MSK does MSK Impact and Stanford does their thing. They don't do RNA. And then you need to do some special extra thing to get the information about targetable, even the hint of a targetable protein.
Yeah.
So I think that there's going to be increasing pressure towards surface these targets, and create that candidate list in a higher throughput way. And then, do some sort of validation in a higher throughput way. You can't be limited by, the path lab has no antibody for this thing that might save your life.
Yeah. So if you're talking about, you're talking about this is a problem suitable for a FRO. This is really a problem. I mean, imagining a clinical trial where you take, 200 cancer patients. It's a big basket trial, and you randomize them into a bunch of molecular studies for screening, CLIA validation of hits, return that information to the patient and the treating clinician versus, standard of care to 100 patients. And you just follow them out 5 years.
Yeah.
And then you answer your question, you see, doesn't matter.
Yeah.
Does this additional information actually lead to more effective therapeutic decisions for the patients and the clinicians? Or was standard of care just been fine all along?
Do you have a suspicion on which way it will go?
I definitely think more acting on more molecular testing.
I think that 10 years ago wouldn't have made a big difference. And there were trials like this. There was one big trial trying to do precision oncology with a really limited set of therapeutics. And they had stuff BRAF inhibition with subclonal BRAF mutations that ended up not mattering in some cancer. They're like, oh, precision oncology doesn't really work in 2015. I think if you did it now, it would be dramatically different.
I remember I was looking through Sid's website, and like he had like he did single cell RNA-seq, and I was like, oh, that's normal, right? And then I asked like an oncologist on her team, and then she was like, no, like no one ever does
Sid,
that.
Sid has beautiful data. He's got
Yeah.
a bunch of single cell long read RNA sequencing, and it like him doing that and a few other Pathfinder clients doing it completely won me over. That as like the one true assay. It's really all you need.
Yeah.
You get the T cell receptors of the TILs. You can see the mutations in these long transcripts. It's like you just had to do one assay. That's the one assay. And it's almost never done.
Yeah.
Like a handful of billionaire cancer patients have done it on their samples.
Like there's no like diagnostic test that requires that, right?
No. So if you wanted to return it to clinicians in the classical fashion, you would have to get that whole thing CLIA validated, right, in some lab with no actual biomarker case for funding it, right?
Just information.
just for the fun of it.
We're almost at time, but like, I want to hear like a short paragraph from each one of you. If you were given $100 million, what would you do to push this field forward as fast as you possibly could? Go first.
Okay. I really want to do within patient controls and clinical trials. I want to run clinical trials that give you maximal information about your design decisions, and clinical trials kind of cost a lot, so that you can spend $100 million on that. I think specifically the boring version of that is things that involve vaccine platforms, so like immune responses. That's not the really interesting part. I think the more interesting part is doing targeted mass spec validation, quantitative immunopeptidomics on all the things that you put in the vaccine from different selection methods. So you take your like algorithmic signals that currently you just kind of like bake in a couple things based on your priors with no real validation, and you fill the vaccine from multiple ranked lists. So take 2 ranking methods, whatever those in silico signals are, fill a vaccine with both of them, and then you have the patients. They're going to have a resection. You can get a lot of tumor tissue, and then spend a bunch of money making the heavy labeled peptides and confirm which of those antigens are actually in the tumor. Ideally, also do it on some antigen presenting cells from the patients, and like work through the whole chain of causality. I vaccinated them, their immune system presented these peptides, their tumors also presented these peptides, and then they had strong ex vivo responses against these particular epitopes. And that's why the vaccine didn't work, because the 2 immunodominant ones were not the 4 that lined up between the APCs and the tumor. I think that because they're trials, they would, burn through a lot of money that way, but it would teach you stuff that like hundreds to thousands of trials that don't do that do not teach you.
I have questions, but I'll ask you them later and then put them in the text of this video. What about you?
So I don't disagree with Alex at all, but assume I have my own $100 million.
Yeah.
I would set up in addition to things like that, a mouse translational system where the animals are essentially wild and are carcinogen exposed, but also had complete control over experimental design. And then I would create a clinical trial context for testing complex immunotherapy strategies and optimize vaccination, TCR-T, immune checkpoint inhibition, combinations, but in ways where, you have all access to tissue and so on and so forth and can understand.
This feels like something you could get a grant for. It feels very NIH shaped. What's the?
I haven't tried, but and so maybe, but it would cost a lot more than $2.5 million over 5 years for one R01, and I don't honestly know what I would learn. Right?
Yeah,
I have 20 hypotheses about what therapies and therapeutic strategies would work best and why, and I could test them all in parallel if I had hundreds of mice being enrolled every month. But if I have to just do one track at a time, then it almost becomes not worth it.
that makes
Okay,
sense.
so I came up with a different one. So the one I gave you is sort of more the one I've been kind of incubating in conversations for a long time. So that's kind of my go-to, like, oh yeah, we should profile everything deeply, figure out causality. This conversation about like the founder mode cancer trial that's not limited to cancer vaccines, but you do high throughput profiling on, take half the patients, they get standard of care, do high throughput profiling on the other half. And ideally, you could structure it so it's not just a suggestion to the treating oncologist, because that's kind of the way to make it acceptable to the existing system, but maybe have actually a shared platform where you make therapeutics for them, like RNA encoded therapeutics against a wide variety of targets. So try single platform, informatically selected target and therapeutic. And then see if that is like, can you make the box? Like literally like tumor tissue goes in, profiling happens, target selection happens, therapeutic selection or design down into an RNA sequence all happens as an entirely encapsulated computational process. And what comes out is synthesized RNA.
Yeah.
I think if I had $100 million, I would want to try that. Yeah.
I'd want you to try that too.
But it wouldn't inform the cancer vaccine stuff we've been talking about.
I don't think we know enough about how to overcome suppression and evasion in the tumor microenvironment to do that in a personalized way. And I think we're just going to bump up against the ceiling of cures until we can personalize those things just as much as we can personalize TCRs or vaccines there.
Yeah, this isn't like, I haven't deeply considered it. It just sounds exciting. It's like, can you take the founder mode spirit and then like put it into such a streamlined, automated fashion that it's all one process?
In some sense, it feels like that is about to happen with the for-profit companies that are spinning up, like Valius. Like that's going to be, is that not going to be a natural experiment they're basically running? Like people who,
I mean, I like, we're in the Pathfinder camp, but like Ed Larkin, the head of Valius, I like him a lot. I've talked to him a bunch, and we've interacted with the private health people who are the sort of the, slightly less high tech, but still like in the same space. And I don't think that anyone has some real secret edge there as far as how the analysis happens. And it's very manual.
Yeah. I think the key point to your $100 million dream though, Alex, was not just identifying targets. Identifying targets is hard, but that's really only the first step.
Yeah.
You have to be able to action the targets in a way that's beneficial and non-toxic for the person. And throwing a bunch of genomics data through some computational processes to predict a set of possible targets, that's doable now. Yeah. But actually figuring out how to action all the targets in good ways, like cracking that is...
Yeah. I think what currently happens in this like hyper concierge context is that you facilitate access to a bunch of high throughput profiling. You get all the data, you run a bunch of tools on it, someone's doing that. Then they've got their Jupyter notebooks full of analyses and they're making figures. And a few weeks later, they have a slide deck. That slide deck goes to the client who then like talks to their doctor who's skeptical. And then eventually you talk to the doctor and then you make a new slide deck for the doctor. And then eventually you're kind of nudging a decision towards like, yeah, you should try that TRP2 ADC because there's a lot of TRP2 expression. This like takes like a few months and maybe you get them access to something that they could have gotten access to before. What I'm talking about is like, why don't we try the extreme form of that in which all of that is being automated?
Like the doctor no longer, like the patient and the Plus platform is all the agency that goes into managing the cancer.
Yes.
There is just no like institutional presence that's like watching over.
Yeah. It's like you're like, last line of defense against a cancer.
Yeah.
You take someone who didn't have a great option and then you input tumor sequencing or even, whatever. It's like profiling. And then try to push it through further than just find a good target to also, we know that there's this antibody for that target and we can conjugate that to an anti-CD3 domain. And that could be a bispecific that's a T-cell engager for the target on your tumor. And we know that we have a shared way to encode that. We can make an mRNA encoding of it. There's been a few trials of mRNA encoded bispecifics. And so that's going to be the therapeutic that this box gives you.
Mm-hmm.
And so you skip the like, me making figures in Jupyter and then assembling the PowerPoint to convince a chain of people to then ask for access to something. You just say like, we find the targets, we make the therapeutic, here's the therapeutic.
I kind of assumed like Valius was that. Like it has a therapeutics. Okay.
It is not. And we try to put one inside of Pathfinder through this TCRT thing, but making TCRTs is really slow. The version of this that is like, data comes in, therapeutics come out does not exist yet.
Okay.
Yeah.
That's a
And I mean,
fantastic.
and ideally, what you'd have at the end of the day is your box would give you, here are your 7 options. Here's everything I can know or predict about potential efficacy and toxicity profiles. So you go and you speak with your clinician, you have that goals and values and deep discussion and explanation back and forth and everything, and then you make a plan. But you have it all there for you relatively easily, broken down by, here's 3 things that you can get off label. Here's 1 clinical trial. Here's 5 things that the box can actually make for you if you want that.
Right. And, one of those things is like the expression value is lower, but it's very tumor specific. And this one's HER2, which might have cardiac toxicity risk.
Right.
So I think that you're right. There's a slightly more complicated package to deliver, but I think the key thing that does not at all exist right now is an end-to-end process that goes from data to therapeutics.
Yeah. And personally, I would really love to practice that. I wish that box existed and it would drop in my clinic. And so a patient would come and that's what my interaction.
I feel like I could ask questions for another few more hours. You guys are incredibly fascinating. But unfortunately, I do have to catch my flight out of North Carolina. Thank you so much for coming on to the podcast.
Thank you for coming.
Yeah, thank you for having us.
Yeah, that's awesome.